There's A.
The green stuff. Oh, no. This one is the right side. Have you ever seen this? It's like hello, good morning. How are you doing? Oh, you draw it like fours. It's like that. How are you doing? I can find out what you have. I made so much food.
If you can squeeze a little bit. Looks like we are.
You're okay. You're okay. You're okay.
One announcement. The report that I was talking about is the final report. It's not for some students were asking whether they need to write a report for every lab. No, they don't need to do that. It's the final report at the very end of the final.
We'll give you guidelines.
We're going to print out the guidelines and hand it out maybe tomorrow, today, or later today.
Morning, everyone. We're going to get started. We give Scott as much time as he can possibly give him. He's going to do the impossible this morning. Completed an entire picture of cell biology for us, right? So Scott Season's going to do this for about 15 minutes, and then we'll have time for questions. So please hold your questions until that time.
Morning again, folks. I'm glad you all decided to go ahead and show up again today, even after yesterday. I hope this is helpful. But we're really only beginning. And remember that the four major classifications of biological molecules that we're talking about, well, that we talk about in general, are lipids, which we covered yesterday, in terms of the way they help to make up the membranes that define and surround and maintain the cell shape. But also within a eukaryotic cell, these phospholipid bilayers also set aside different compartments within the cell to hold different functions, such as the nucleus to maintain our DNA, such as the Golgi apparatus where we process some proteins, such as, well, several other organelles that carry out different functions. Then we started talking about nucleic acids, and we didn't even get through nucleic acids yesterday. So we're going to continue talking about the roles of DNA in terms of carrying the information from generation to generation. And then we're going to get into a little bit of how RNA helps DNA get turned into protein. Okay? And then we're going to talk about protein. Unfortunately, we're not going to talk much about carbohydrates, right? Sugars. Sugars play a wide range of functions in the cell. but we're not going to have time to really cover that much at all, especially if I want to do any justice whatsoever to proteins. So this concept of how we turn a DNA nucleotide sequence, remember our A's, G's, T's, and C's, into a protein is a two-step process. The first step is called transcription, rewriting, and the second step is called translation. Overall, these two large steps together comprise what we call the central dogma of modern biology. I think most scientists dislike the idea of dogma, right? We'd much rather have evidence than dogma, I hope. This is the whole point. But this is such an important central part of this that we call it the central dogma. And actually, I read some things by Francis Crick lately that he had a much more narrow definition of the central dogma in mind when he coined that phrase for this. But we've come to mean these two things and how we turn DNA into protein. Okay, so the idea here is transcription, rewriting, is the first step, and it's how we are going to turn, again, this DNA nucleotide sequence into an RNA nucleotide sequence. Notice in both cases we're talking about nucleotides, polynucleotides, just DNA and RNA. That's why this is transcription. We're transcribing basically into the same language. We're just making a copy, right, of this original sequence. Okay? So I think if we can get the lights down a little bit, that might be helpful. I've got a little spiffy animation. Oh, I hope this doesn't try to reload it. I've got it already loaded and ready to go. Let's see here. How did I do? Here we go. Must be this one. All right, so this is transcription. I'm going to show this little animation. Basically, this guy describes it better than I could. When we're done, we'll go back and we'll go through it step by step. So don't worry if some of the details fly past too fast. We'll come back and get it covered again, okay? Oh, somebody asked if any of this would require audio. Actually, this would be helpful at this point. Can this happen? Is that easy? If not, I can...
It's running audio and running
Okay, cool. Cool.
So you could
Put the microphone against.
Ah, very good.
One of the DNAs.
We'll start this again. Perfect. Thank you.
RNA polymerase first separates the two strands of DNA, and RNA nucleotides present in the nucleus bond with their complementary nucleotides on one of the DNA strands.
Okay, I have to stop this guy. I interrupted him already, right? So we did mention briefly yesterday, I should have reminded you before I started my animation, that, that this green blob here represents a complex of proteins that we call RNA polymerase. It's making an RNA polymer based on the DNA nucleotide sequence here in the double-stranded DNA. Now, it's only going to copy one of the two strands. Now, when I say copy, what do I mean by copy? Remember our base pairing rules that we talked about yesterday? A always binds with T, and G always binds with, right, except for RNA where we don't have T's, and A binds with U. Very good. So that's exactly what RNA polymerase is doing for us here. It's using those base pairing rules to bring in these RNAs, nucleotides, and base pair them up. So if this is a T, right, it's DNA, so it could be a T, then this one's going to be a U. If this is an A, this is going to be a U. If this one's a G, this is going to be a C, and if it's a C, it's going to be a G. So that's how it knows which nucleotide to bring in to make this copy of this strand of DNA. Questions about that? All right, now I'm going to try really hard to keep my mouth shut and let this guy talk a lot. This really is good.
Remember that G and C bases always bond together, and the A and U bases of RNA always bond to the T and A bases of DNA respectively. RNA polymerase then links the RNA nucleotides together to form mRNA in the process called transcription. RNA polymerase begins at the promoter site and then advances into the gene, separating the DNA double helix at its front end, joining together the RNA nucleotides in the middle, and then rewinding the DNA double helix at the back end. When the gene is entirely copied, the RNA, DNA, and polymerase then dissociate from one another, and the newly formed mRNA is released to go on to the process of translation.
Okay? So this is the first step of a two-step process. Transcription, we're just making a copy of our DNA into RNA. This then leaves the nucleus to go out into the cytoplasm and find a ribosome to carry out translation. Questions? A bazillion, right?
So, does the RNA polymerase know what kind of nucleotide is coming, or just spontaneous finding?
It has no idea what's coming. All it does is it allows an RNA to come in. Let me back up to the point where this is actually happening in the animation here. And this doesn't get covered, and it doesn't get covered very often. But all it's doing is allowing these RNAs to come in, and if they bind tightly, see, this is the thing, A and U bind with each other pretty tightly, more tightly than A and G or A and C do, right? So if this is an A and a G comes in, they don't hydrogen bond with each other very nicely. It's a special kind of chemical bond called a hydrogen bond that holds these things together, and it'll have a couple of them. But anyway, it won't bind right, and RNA polymerase will just let it float away again. If a U happens to come in, which is what it's wanting to bind with, that happens to bond pretty nicely and becomes stable, and RNA polymerase goes, oh, that's a nice stable base pairing, make that connection between this one and the next nucleotide. Okay, good. Excellent question, yes?
RNA polymerase, how to choose the directions of the synthesis from because DNA has two substrates.
Exactly. And who's to say it's not going to go on this strand and go this way, right? Sometimes it does. Here's the thing. There is a special region of nucleotide sequence called the promoter that's on this strand that needs to be copied. And there are several different types of promoters. There's a cat box and a Tata box and several other types of things, just regions that RNA polymerase recognizes, or some subunits of RNA polymerase recognizes, hey, this is a good place for me to sit and start to transcribe this gene. And if that sequence is here, it'll go this way. If that sequence is here, it'll come this way on the and do the other strand. You're starting to see why I can't possibly cover everything in 3 hours. Because I literally can spend a week talking about nothing but promoters and how they bind with RNA pol2 and how that gets regulated, and it's critically important. Okay? Keep thinking about it. I'm more than happy to chat with anybody. By the way, I'm sorry that I wasn't able to stick around yesterday after my talk. I do plan to be around at least through lunch today if anybody wants to grab me and berate me for the horrible job I did, or ask me questions or whatever. Okay? Am I good enough to keep going?
Okay.
If there's something else you can ask, I'll be happy to talk a little more about it, but we've got to move on eventually. So ask, if you got one more thing, ask.
Okay. Does any possible have the two promoters in the same region at a different
Yes.
substrate?
That does happen a few times. So we used to think one gene, one protein, right? But that gets a little complicated. It's possible that somewhere in the middle of this gene, the other strand has a promoter region that would read a gene in that direction. And this is getting really complicated now, how all that gets regulated, but it does happen. Nothing is ever as clean and clear as we hope when the further we study into biology. Good, good. Anything else? Yes?
You know how DNA bond with the RNA
Yes.
If you go from the other strand, wouldn't it be 3 prime?
So that's why I was saying it would be on the other strand and it would go that way, right? This one is moving this way along the DNA because it is moving from 3 prime, right? That free 3 prime OH is what gets added to. But if it was on the other strand, then it would of necessity be going that direction because it's always going to, so the 2 strands, I wasn't going to get into this, the 2 strands run anti-parallel, right? So if this is 3 prime to 5 prime on this one, this is going to be 3 prime to 5 prime on that one. Okay, excellent questions. All right, good. So this is basically just showing what we had in the animation, and we've got this region of DNA just before the gene of interest here that's called the promoter. This is where RNA Pol II originally sits and binds and says, okay, I'm going to start to transcribe the gene that's here. Okay. Comparison, these are crystal structures. X-ray crystallography is used, to show atomic level detail of how these proteins look. And look, all the way from yeast, which is basically the simplest eukaryotic organism I know of. It's often a single-celled thing, a yeast, and it's got a nucleus, and it's eukaryotic in every way, but it's very simple. All the way since then until, well, from bacterial till yeast, right? Huge difference in evolutionary time. Look at the overall similarity of that structure of RNA polymerase. It's a really critical function. It's necessary that it has very much these specific shapes to give it its function. So that even in us, humans, hasn't changed significantly from this overall shape. Now, we'll talk more about why this looks so messy in a little bit. This is really, well, this is quaternary structure, but any one of these colored regions, like the yellow here, is one long strand of protein. Okay? The dark blue here is another one long strand of protein. And they all fold on themselves to give these specific structures. So RNA we often use to tell us, this is a technique called in situ RNA hybridization. And what we do is, if we have a particular gene that we're interested in knowing about where that gets, what cells make that gene, we set up a probe for the RNA, for the mRNA, for that gene. And we can stain, in this case, these are Drosophila fruit fly embryos. Okay? And they have stained these for a particular gene. In this case, the name of the gene is called hunchback. Right? And early in the embryo, that mRNA is being transcribed here and at the very posterior in here. Later, it comes in these little bands. Then it gets even more specified. Those cells doing that tend to migrate and get themselves into look at this very specific little pattern. Just these 2 cells here, 1 or 2 cells here. Those cells are expressing this and none others. So it's by looking for the mRNA, we can get a really good idea of where in this embryo we are expressing this gene, where we are transcribing this specific gene. And we can set up a probe for any gene you happen to be interested in looking into. Okay? In situ hybridization, very useful technique. And the point here is, as I said yesterday, all cells have the same set of DNA. So my blood cells and my neurons Red blood cells are a bad example because they kick out their nucleus, right? They don't have DNA anymore. But my muscle cells and my liver cells and my pancreas cells and all of these cells have the same DNA, but they've got to take on very different structure and function. Right? And as I said yesterday, if lipid was all there was, they'd all just be round bags of water. Right? But the proteins that they express give them their different shape and function and structure. Okay? Questions? How does it know which genes to express in which cells? How does my heart cell know it's a heart cell and it needs to express the genes for that type of muscle? And my eye cell knows it's an eye cell and needs to express those different genes. I'd be happy to talk to you for about 3 weeks about that. What it comes down to, the main method by which a gene gets turned on or turned off is at the level of transcription. And there are proteins called transcription factors which go and sit on that promoter region, remember the promoter region of DNA? And if they want to activate a gene, they literally recruit RNA polymerase and say, come, sit here, transcribe this gene, go. Right? If a gene is not supposed to be expressed in there, like there's a particular gene for a pigment that's in your eye, that your heart doesn't need that pigment, right? We do it no good. So there are other transcription factors that go to the promoter of that gene and say, RNA polymerase 2, go away in this cell. Leave me alone. You don't need this gene. Don't bother wasting the energy to transcribe this gene. It wouldn't do us any good, right? So by doing this, and it's very complex, still being starting to be understood, system of saying, I need this set of genes in this cell, and this overlapping but not identical set of genes in this cell. Okay? And they're called transcription factors. We'll talk a little more about how they get activated as time later on this morning. All right, so that's transcription. Now we're going to talk about translation. In other words, we're going to take that mRNA that we just made in the nucleus, it's going to leave the nucleus and go out into the cytoplasm where it's finally going to make us our protein. It's going to make our protein for us. Okay? And the structure where this is going to happen is called the ribosome. Every living organism on the face of the planet uses ribosomes. With RNA polymerase 2, I had no problem calling that an enzyme that catalyzed transcription. But when we talk about the ribosome, for technical reasons, we are a little uncomfortable calling it an enzyme. Because the technical definition of an enzyme is a protein that catalyzes a chemical reaction. And ribosomes are really interesting structures because they're a combination of protein and RNA. Don't worry too much about that. But because they're not strictly protein, we are a little uncomfortable calling them enzymes, and we just call them the ribosome, a structure. We come up with these other ways of saying it. So the site of translation is a ribosome. I've got another animation to show you about this, and then we'll talk about it quite a bit more. Well, let's see. So that's this one. All right. So here we are. We've gone ahead and we have generated our mRNA, which is this whole long thing. It has left the nucleus. It's going to find its ribosome. And that ribosome is going to read this RNA, this mRNA, in sets of 3 nucleotides at a time. Okay? 3 at a time. 3 at a time is called a codon. And every codon, C-O-D-O-N, is going to code for 1 amino acid. Okay, I promise to tell you more about what an amino acid is in a couple minutes. A lot more than you want to know. Okay? But it codes for 1 amino acid. And a protein is just a long chain of stuck together amino acids. Okay? So by coding for the amino acid sequence, the mRNA codes for the entire protein. Let's watch the animation. Let's talk a little more. Feel free to ask questions. Okay?
mRNA is used in the process of translation, or protein synthesis, which occurs on the ribosome.
Purple blob
The
of the
ribosome is positioned so that each of its 3 binding sites aligns with the codon of the mRNA. Transfer RNAs, or tRNAs, carrying amino acids approach the mRNA. tRNAs with anticodons complementary to codons of the mRNA will bind. When the middle and first binding sites both contain tRNA, the ribosome attaches their amino acids together. The ribosome then moves down the mRNA the distance of 1 codon, so that the tRNA in the first binding site is now in the middle binding site, and the first binding site is available for another tRNA. The tRNA that has lost its amino acid is moved to the last binding site and released. This process continues, with tRNAs bringing in new amino acids, binding their anticodons to the codons of the mRNA, and having their amino acids added to the growing protein chain. The process stops when the ribosome reaches a stop codon. This codon does not bind with any tRNA. Instead, it signals to the ribosome to stop translation and release the finished protein.
Okay. So we've got our mRNA down here that brings the message from the nucleus. Then we've got another type of RNA called the tRNA that brings in the amino acid. Now remember yesterday when I was drawing an RNA, I said sometimes that RNA can fold back on itself and base pair with itself. One region of the molecule base pairing with the other region. It's exactly what's going on to give these tRNAs their shape. And when they fold on themselves correctly, they've got an amino acid stuck to one end originally, and at the other end is a 3-letter RNA sequence called an anticodon. Okay? So remember how in RNA Pol 2, it waited until there was good base pairing before it added that nucleotide to the growing RNA. Right? Same thing happens here in the ribosome. It waits until the correct tRNA comes and nicely base pairs with the mRNA before it catalyzes the addition of that amino acid. So this one is GUU, and so its complementary anticodon is CAA. If one that reads GUA tries to bind here, it simply won't base pair and it'll float away again, and its amino acid won't be added. But when a correct tRNA with the right anticodon and therefore the right amino acid comes in and binds, then that amino acid gets added to the growing chain. If I had my druthers, that would be the end of the lecture for today. You could go home and think about that all afternoon and come back before I go on to the next thing. Right? Yes?
So what is the tRNA? What's the amino acid?
It floats away, and it goes off to be recharged with the amino acid again. Yes? Yep, they get recycled, yes.
Are all the ribosomes essentially the same?
Yes. All ribosomes are essentially the same, whether we're talking about bacteria, or trees, or meat. Some ribosomes are associated with some of the membranes inside the cell called the endoplasmic reticulum. They are making proteins that can be embedded into the plasma membrane or excreted from the cell, and they're exactly the same as the ribosomes that are floating free in the cytoplasm. Ribosome is a ribosome is a ribosome. Yes?
So when the tRNA is getting recharged, what's the mechanism by which the amino acid recognizes the tRNA to get attached?
It's a great question. This is kind of a gap in my personal knowledge. A lot is known about it, okay? I have neither the expertise nor the time to really give a detailed explanation of how that works, okay? But needless to say, the enzyme that does catalyze that charging of the tRNA with the amino acid is very specific. The anticodon determines very precisely which amino acid gets added to it. It's a great question. Please look into it and email me and tell me the answer. Okay? Yes?
Do we know how each amino acid connect to each other?
Yes. We very precisely, and I promised to get into that. His question was, do we know how one amino acid attaches to the other? And yes, we know very much about that, and I'm going to talk about it for the next few minutes. Okay? It's called a peptide bond. Okay? And we'll talk a little more about that. Okay, so these are some different views of tRNAs once they folded up on themselves. This is that type of thing we just saw. It's just a stick figure. Look, this very much emphasizes the areas where these bases here bond with each other within that one molecule. Right? They base pair with each other to give this its structure. This is more of a crystallography determined shape. This is how they really kind of, the RNA folds up on itself to look like this. Notice here again, you've still got this anticodon loop. How many bases will make up that anticodon? 3. Exactly right. Okay? And this is more of a space-filling molecule where it's actually given a kind of a 3D representation of all of the atoms in a tRNA. And then this is a nice little icon that we can use to really get the idea across quickly. Okay? But this is what those tRNAs actually look like. All right, so here again, we've got the 2 subunits of the ribosome. Notice when they come together, they sit very nicely with the mRNA going through them. And these 3 binding sites for tRNAs. One common misconception would be that you might have a ribosome sitting on an mRNA making its protein until it's done, and then another one binds. But it's entirely possible for many, many ribosomes to be sitting on a given mRNA, simultaneously translating lots of copies of this same protein. If the cell needs a bunch of this protein, this can very easily happen. And here's some electron micrographs. Look at all these RNAs bundled together on this mRNA. I'm sorry, all these ribosomes bundled together on an mRNA. Apologize. These are ribosomes sitting on an mRNA. So this could be making a lot, just churning out a lot of these proteins. Notice that here it's just started. Right here it's a little longer. We've added a few more amino acids, a few more amino acids, a few more amino acids. By the time we got here, that's a nice long protein that folds up on itself and goes off to do its function. Okay, they are moving along, ratcheting along that mRNA, churning out protein. This is called a polyribosome. Again, just to drive home some of these things, we've got, in this case, our amino acid up here is phenylalanine. We've got the tRNA, we've got the anticodon, we've got the codon that's on the mRNA. RNA has 4 bases: A, U, G, and C. Remember that those 4 get read in triplets. If you do the math on this, you can see that there are a total of 64 possible codons. One thing I want to tell you is that we only have 20 amino acids that we use. A few others here and there throughout the living world. If you get those 20, you're just fine. Okay? So 64 possible codons, right? But only 20 amino acids. What does this mean? This means that there are, then there are 3 codons that get read as, hey, you're done making this protein, kick it out, right? Good job, well done, we're finished. They're called stop codons, all right? So this leaves us with 61 codons coding for 20 amino acids. By the way, there's also a start codon, AUG, that calls for one of the amino acids, methionine, but it's also the one that says, hey, ribosome, start translation here. Okay, so we won't get too much more into that. The ribosome basically scans along the mRNA until it finds an AUG, and that's how it knows this is my first codon, and this is the beginning. So every protein starts with methionine, because AUG is also the start codon. All proteins are built at the start codon, starting at the start codon, so all proteins begin with methionine. All right. Here's the thing. There are some codons that multiple codons call for the same amino acid. Okay? So we've got 20 amino acids. By the way, we know this codon, we know the code. We know what codons call for what amino acids very precisely. Okay? So look at this. CCU, CCC, CCA, and CCG all call for proline. Any one of those codons tells the ribosome, bring in a proline, and proline is the next amino acid in this chain. Okay? CAA and CAG both call for glutamine. This property of this code is called redundancy. There are multiple ways of getting threonine as your next amino acid. At least 4 of them. Right? 4 of them. These 4 codons. This is one reason why it's not like a blueprint and you can't go backwards from protein to RNA, because you don't know which one of these 4 called for that. So the genetic code is redundant. Right? There are multiple ways of calling for a particular amino acid. However, the genetic code is not ambiguous. What in the world are you saying? Redundant but not ambiguous. What does that mean? While it's true that there are multiple ways of calling for one amino acid, it's never the case that a codon might sometimes call for proline and sometimes call for lysine. That never happens. If you have this codon, you know for a fact it's going to call for threonine every time and never anything else. It's not ambiguous. Okay? So the genetic code is redundant. There are multiple ways of calling for alanine, but in no case is it ever ambiguous? If you have GCC, you have alanine every time, never anything else. You see the difference there? Questions about that? Yes.
Why tryptophan has only one codon?
Great question. Evolutionary history of how the ribosome is formed. It's only got the one. I don't know. It's a good question. Yes?
Isn't that also dependent on like the, you know, the mutation issues? Like like certain things cannot be mutated, like proline gets mutated, it changes the whole structure, which is why all four, any of the last changes can lead to only proline again.
That might be part of why the code is the way it is and how it developed over time. His point is, with proline, look, if you mutate that third base of the codon, it doesn't matter which one you mutate it to, it's still going to call for proline. Right? Whereas with, what were you saying, tryptophan? Show me where it is. I can't even find it right now.
Upper right.
Upper right. Okay, yeah, tryptophan. If you mutate this codon in any way, shape, or form, you're now going to call for something different, and you're going to have a different protein. Okay? So this type of mutation, where we go from ACU to ACC, in both cases, your protein is exactly the same. You've made no difference in that organism in how it functions at all. We call these a silent or neutral mutation. Very good. One last point about this genetic code. It is redundant, it is non-ambiguous, and here's the kicker, it is universal. Every single organism alive on the planet today uses this exact genetic code. Whether it's a bacteria, or a rose, or a fungus, or me, we all use that exact genetic code. This is why genetic gene manipulation is so powerful. The possibilities are so exciting, and at the same time, a little bit scary. Okay. So let's practice for a minute, just for giggles. The way this readout, and I've seen several different representations of the genetic code. This actually isn't my favorite, but it's one. If I said this is an mRNA, and I want to translate that, by the way, I talked a lot about the word transcription, rewriting. I didn't talk about the word translation. When I'm translating something, what am I doing? Going from one language to another language. When I'm transcribing, I'm just copying, right? Nucleotide to nucleotide. When I'm translating, I'm going from the language of nucleotides, mRNA, to the language of amino acids to make a protein. Completely different type of molecule. I promise to show you the amino acid very, very shortly. Okay? So it's translating into a whole different type of biological molecule. Nucleic acids to protein. All right, so if I want to translate this into an amino acid, what's the first thing I'm going to want to do? Good answers, whatever they were. I'm gonna break it up into codons, right? And I'm gonna my ribosome is going to know AUG is going to be my first one, and then GGA, GUC, so forth. To read this particular type of chart, what you'll do is you will look. Notice this says first base. We're not talking about dating here. First base. You look at your first base, and it's along here, so it's A, so I know it's going to be somewhere in that row. Okay? Second base is along the top. U is right there, so I know it's going to be in that column. So where those intersect, it's going to be somewhere in here. And then I look at my third base, and that tells me that it's going to be methionine, which makes sense because it's the first one, and it's going to be my start codon. Right? So if I want to do my second one, right, it calls for methionine. My second one then, GGA. I'm going to look at my first base. It's going to be a G. So I know it's going to be somewhere in that row. It's going to be another G, so I know it's going to be somewhere in that column. So no matter what that third base is, I know for a fact it's going to be glycine. Right? Somebody want to do the next one? What's it going to be? Say it loud. Thymine? Valine. Uh-oh, how do we go wrong? All right, G, U, so it's going to be in this row and this column, and so no matter what the third one is, it's going to be valine. Work for yourself and see if you can get it. Good, threonine. What are we saying here? Tryptophan. Okay, good. And glycine. Okay, GGB. All right, so that's going to be my amino acid sequence. Yes?
Shouldn't the last one be a stop codon? Do we need to have that?
If it was a full protein, then yes, you're right. The last one would have to be a stop codon. Yes. Dot. Yes.
If any one single base is missing in this sequence,
Ha!
the protein will totally wrong.
Exactly. That is called a, let me repeat what you just said for everybody, okay? He says if any one base is actually removed, every single codon from then on will be completely wrong. And he's got it exactly. Also, if you add a base, right, then everything from there on is basically going to be, you're going to shuffle the deck and you're going to get random amino acids. This is called a frame shift mutation. I'm sorry, keep asking. That's a great point.
How to prevent in the natural
By having the bonds between these nucleic acids be covalent, be stable, trying to look for, and the cell has a lot of ways of looking for, remember, back in the DNA, this is RNA, right? But back in the DNA, this was double-stranded. If you remove any one from this strand, the cell can tell this base pairing no longer is right, something happened here, and it usually will just kill the cell, right? Because we can do without one or two cells. Yeah, excellent points. Other questions? Yes?
Do you know if there is any mechanism to remove the start codon and the stop codon from the proteins, or all the proteins starting?
Okay, the stop codon doesn't actually code for an amino acid. Okay, so it doesn't add one that should be removed. The start codon, my understanding is that really every protein starts with the thymine. Biologists, am I wrong about that? Does it get truncated? Does it get pulled off?
Sometimes it depends.
Sometimes it depends. I think most proteins really do start with the thymine. Now most proteins are hundreds or thousands or even tens of thousands of amino acids long, so one amino acid probably isn't going to screw anything up. Okay? Do you know of any specific examples where it gets pulled off?
I know when we do it, like when we're doing, we're taking a particular gene and putting it into bacteria, we ensure that the thymine gets truncated.
Okay.
We can do that.
Okay, because you don't want it starting in the middle of the gene.
Right.
Right. Yes? So I think that's what I meant, if I understand the word synonymous there, that's what I meant by a neutral or silent mutation, right? So you change a base that is redundant in the code, and it calls for the same amino acid, and that makes no difference in the overall protein. Do I understand that term correctly? Okay, good. Yes?
Is any explanation for why the stop codon is always
The best explanation I know of is simply that the ribosome needs some place to tell it what the frame is. Right? So if you didn't always use the same AUG, right, then it wouldn't know where to start. But the ribosome uses, it actually starts at the first beginning of the mRNA, and that first bit of the mRNA may well not be translated. It scans until it finds an AUG, and it says, okay, not only now is this my first codon, but that's where I set my frame. The first time it finds this sequence, AUG. Now there may well be other AUGs within it, and they'll just call for methionine. Okay. I know. All right. Finally, this is an amino acid. And ignore the green blob for a moment. Just look at the chemical formula there. And what you can see is there's this central carbon. Then on this side, there's an amino group, basically a nitrogen and 3 hydrogens. On this side, there's a carboxyl group, a C double bond O. Every single amino acid, that's the definition of an amino acid, is that you have a central carbon with an amino group here and a carboxyl group here, and then some side chain. Don't let yourself think that side chain means unimportant. We're going to see how critically important those side chains are. But an amino acid has the same fundamental base, and then 20 different side groups to give us our 20 amino acids. Okay? Did anybody play with these blocks when you were a kid? My daughter has these blocks, and that's what we're trying to represent here. Okay? So notice that every single amino acid has this same region here. So they've all got this little blip here, this little nipple here. And every single amino acid has this same carboxyl group on this end, and that's this little hole. They all have the same, right? So they all have this end the same and this end the same, but their side groups are very different. The important thing about this is that the amino terminal of one amino acid can bind with the carboxyl terminal of another amino acid. Okay? This is just a normal, average, everyday, unimportant, covalent bond, like any other, but this joining of one amino acid to another is so important to biology that we gave it a special name. Anytime we join one amino acid, the amino terminal of one amino acid to the carboxyl terminal of another, we call that a peptide bond. So this represents a peptide bond. What the ribosome does is bring in methionine, whatever comes next, whatever comes next, so forth and so on. You get the idea, building up long chains of amino acids. Okay? And if you're my daughter, you can't stand, whoops. You can't stand to have them all together. You got to pull them apart, all right? So, this never happens. That doesn't happen, right? It comes in one at a time in the ribosome. But when you're all done, you can have a long chain of the 20 different amino acids. Okay. Now, notice these different amino acids have different side chains. They've all got the same C and N terminus, carboxyl and amino terminus, but they've got these different side chains. These side chains interact with each other. Some of them attract each other. Some of them repel each other. Some of them are hydrophobic. Some of those side chains are hydrophilic. Some of them are charged. They've got different functional groups. And I'm going to show you a little more technical level of all the 20 amino acids and how we group them in. But the point is, because these side chains interact with each other, they can, this polypeptide, we call a short amino acid sequence a polypeptide, many of these bonds, right? Peptide bonds. These peptides can fold in, hopefully that doesn't happen. Fold in on itself. Oh, one thing I really do want to point out doesn't happen is that. That never happens, okay? That just doesn't happen. But it folds on itself. This isn't really long enough for me to be able to fold, and it looks like this. And because this is the thing that blew me away when I first learned about this, and to this day, it kind of makes my head spin, simply the correct sequence of amino acids and how those side chains interact with each other cause that protein to fold on itself in exactly the right way. We don't have any folding chairs. Pretend I've got a folding chair here. If it's folded up on itself, what kind of function does it have? Really none. It's not very useful to me at all. But if I fold it out, now it's got a function that I appreciate very much, and I can sit on it. The structure gives it its function. In the same way, the structure of how these proteins fold on themselves give them their function. Whether they're going to catalyze a chemical reaction, or be a motor that walks along microtubules, or be a microtubule, or be an actin filament, these are all proteins that attain their function because they fold on themselves in just the right way, and the only thing to tell them how to fold on themselves is the sequence of amino acids and how those side chains interact. We know that this works because we can take an enzyme that has function, that is catalyzing a reaction, and heat it up to the point that it unfolds. Those side chains don't interact right, and it unfolds. And then we can cool it back down and let it fold again. And with some frequency, just on their own, in a test tube outside of a living organism with nothing else but that enzyme in there, they will fold back on themselves enough to give them their catalytic activity back. Yes?
Why aren't a lot of different proteins and pieces of proteins, for that matter, connecting with each other? You know, and why can't it form a loop, or why can't little bits match up with others?
It can't form a loop because the ribosome doesn't bring it back around and do that. It needs a tRNA to bring it in. Okay? And so the catalyzing of creating this covalent bond here has to be catalyzed by a ribosome, and the ribosome doesn't. That's why it can't move back on itself. Your other question, why can't one protein interact with another? It can, they do constantly. I'll talk about that more in a moment. Many proteins, single polypeptide chains fold themselves and then interact with other polypeptide chains to give the pair or the quadruple or the 12 polypeptide chains coming together. They, as a group, they have their function. Individually, they do nothing. Okay? Excellent. Others? Yes? Ha! Excellent. Yes, absolutely. So her question is, you're translating this at the ribosome, and you've got a few amino acids already coming out of the ribosome, but this one down here hasn't even been added yet. Do they start to fold on themselves before they're finished being translated? The answer is yes, they do. And there are other proteins that kind of help them. I have to be really careful about the language I use here because these are called chaperone proteins. And what they do is, as the protein is being translated, they bind and kind of keep it from folding wrong on itself. Right? They give it time to fold correctly. I could talk a lot of time about that, but the answer to your question is yes, they do start folding even before it's finished being translated. Yes. Okay, chaperone proteins help them fold correctly, but many proteins fold correctly within the complete absence of chaperone proteins as well. It's kind of a paradox. Yes?
I've heard of lots of kinds of chaperone proteins.
Mm-hmm.
So, is it the mechanism is finding out kind of non-third people mechanism of showing protein, how to show the protein?
So really, my understanding is all of the chaperone proteins, one way or another, they don't cause it to fold. What they do is they can keep water away from it long enough for it to allow the protein to fold itself correctly. Because if there's water getting there, then that causes some trouble. So these chaperone proteins basically sequester the protein from water until it's time for it to be folded to the interior of the thing mostly. Am I answering your question? Okay. Okay. Yeah. There are badly folded proteins, right? I don't know of any cases where a protein folded one way has one good function and folded another way has a different good function. We do know about proteins that folded this way have a good function and folded this way have a really detrimental function, right? Prions. And so it's definitely possible for proteins to fold incorrectly, and hopefully the cell is looking out for that and keeping it from happening as often as possible. Yes, that also. It can be activated by phosphorylation or something else, and that can change its conformation a little bit, and that makes it active. Yes, absolutely. And that's a major part of cell signaling that I'll talk about hopefully if I get time this morning. Great question. Yes?
And synthesize a protein in vitro?
Yes. Yes, we can. Yes.
Just by amino acid added in.
Add amino acid, add ribosomes in test tube. We get this. We can make this happen in vitro. Yes.
This process needs energy.
Yes, it does. At least ATP, probably maybe some GTP as well, but ATP should provide the energy. Yep, absolutely. All of this requires an energy input. I don't have time to get into cell metabolism and cellular respiration. I wish I did. Again, please, if you're interested in this stuff, please take 3 semesters of biology, or at least one good semester of an introductory class to talk about all of that stuff. Yes?
How these kind of processes are affected by temperature?
Absolutely, very much so. In fact, this is why we as warm-blooded creatures spend so much energy keeping our core temperature very stable. If you get too hot, they unfold. If you get too cold, they don't act right, right? So by spending all of this effort to keep our internal temperature consistent, we can specify that our enzymes, which are proteins that are folded correctly, work within a narrow temperature range. Cold-blooded creatures, snakes, lizards, and so forth, they don't bother spending that, and actually their enzymes have a wider range of temperatures at which they will function. And even if you go outside that, oftentimes, if you then later come back within that temperature range, they will start functioning again.
So the mechanisms are more affected by temperature, or like the proteins and these?
Well, the proteins carry out the mechanisms. So because you're stopping these, or if you get so hot that you unfold these proteins, you can lose that function and they may never fold back correctly, and that's why overheating can kill you. Because the proteins needed for functioning in your brain get misfolded, and now your brain can't work again. Yes?
I've heard about there are not only 20 amino acids and other.
There are a few others.
So it happens in
Yes, yes. Some organisms use a few other, but I'm telling you, as a general rule, if you know about these 20, you're good to go. And if your body, as a human being, has these 20, you're good. Okay? Good. Yes?
And does we know how much calories or synthesis one amino acid.
How many calories for an animal?
Yeah, calories for synthesis one amino acid because it's an engineering problem. We want to know how much energy.
How much energy? I don't have those numbers at my fingertips, right? How many calories they're out there. I will say that however much it is, photosynthesis collects enough energy from the sun to be able to power that. Okay, so the sun is constantly bombarding us with energy. That's how we take that energy from the sun in, is through photosynthesis that produces molecules that the cell can then use as energy to drive these reactions. So basically, you are all solar powered. Oh my goodness. Okay, thank you for the warning. Here are the 20 amino acids. Okay, I'm going to make this presentation available to you guys so you can have this. You can also look it up. But here's tryptophan. We shorten it to that. This is the side chain. Notice, again, the core of every amino acid is exactly the same. Carboxyl terminal, central carbon, amino terminal, and a different side chain every single time. Okay, and these are the 20 side chains of the amino acids. Wow. Okay. I have to talk about this for a second. I can't stop myself. So, as these things are folding, interactions between very nearby amino acids give us what we call our secondary structure. What's the primary structure? Simply the order of amino acids. Valine, threonine, arginine, leucine, lysine. Right? The order. That's the primary sequence. The secondary structure comes from interactions between very nearby amino acids, helping it fold on itself a little bit. Okay? So, primary sequence between my fingers, right? Local interactions between these amino acids give us a secondary, in this case, helical structure. Right? So that's secondary structure. And some of the most important ones are an alpha helix. This is a very common structure for proteins. Or beta-pleated sheets. There's a little bit of beta-pleated sheet up there at the top. Right? This is a secondary structure. Clearly, this is an alpha helix. It's a very common secondary structure. All right? Then there's tertiary structure. Okay? How it folds on itself overall gives it its tertiary structure. And you can see when they fold up on themselves, they take on very specific shapes. This is a channel through a membrane so that something can go from inside to outside the cell. Okay? Then, what was being asked before, you can have what we call quaternary structure. Quaternary structure would be two different polypeptide chains coming together to give the whole thing its function. Okay? So, primary, secondary, tertiary, quaternary structure. Okay? So, general roles of proteins. Chemical, they catalyze, the enzymes catalyze chemical reactions. Structural, extracellular matrix, transmembrane stuff, cell-cell adhesions, signaling, all of these things. I'm so sorry I'm not going to get to talk about stuff that's interesting to you guys, like forced generation of motor proteins, right? Like myosin, kinesins, dyneins. These are motor proteins that walk along cytoskeletal elements carrying cargo. Then there's the cytoskeleton itself. These are very dynamic structures that can give rigidity or motion by polymerizing and depolymerizing. Actin, microtubules, and intermediate filaments. I'm going to skip all of this stuff on cell signaling. Here are our 3 cytoskeletal elements. Joanna, are you going to put this up in your lab, this exact picture?
Not exactly, but very close.
Very close, good. Good, so I don't feel too bad. These are structures, especially actin and microtubules, grow and shrink very rapidly. Okay? So this is the final point I really wanted to drive home for you guys today anyway, is that cells are very dynamic structures. I don't know if I'm going to have the audio here or not. If not, it's fine. I'll just talk.
EB1 is a protein that binds to the GTP tubulin cap at the growing ends of microtubules.
These are growing
Cells expressing a
microtubules in a cell.
GFP-EB1 fusion protein reveal the spectacular dynamics of the microtubule cytoskeleton. Note that many, but not all, microtubules in this cell grow from the centrosome.
Look how dynamic that cell is.
Only the ends of growing microtubules are visible in this experiment. Those that are static or shrinking have lost their GTP tubulin caps and do not bind EB1. In contrast, when all microtubules are labeled with GFP tubulin, the true extent of the microtubule cytoskeleton emerges.
Okay.
Both growing and shrinking microtubules can be observed.
Constantly grow and shrinking, grow and shrinking, grow and shrinking. Now we can make this happen in vitro, but in vivo, this is all such cool stuff. You guys as engineers would love this. I'm so sorry. So the 2 ends of an actin are different, have different binding constants. And one end is much more likely to polymerize, and the other end is more likely to depolymerize. In vitro, the concentration of actin determines whether that whole filament will be shrinking or growing, because they'll be added to both ends down here at these concentrations, or if your concentration is in here, they'll be treadmilling, adding to this side while losing from that side. We can see them actually move in vitro. But in vivo, none of this matters because it's all tightly regulated by other proteins that come and tell actin when to polymerize and depolymerize. Okay? This is what I wanted to leave you with today. Cells are very dynamic structures. When you're doing your mechanical engineering questions, that's fabulous. But even at the level of the cell, remember, you're not dealing with a passive, constant thing. You're dealing with an entity that can sense the mechanical forces and respond to it by expressing different genes. And even at, and especially at the level of the tissue, don't think you're dealing with a passive, non-responsive material. It's something that can, through its own logic, react to what you're doing with it. Really interesting stuff. I'm so glad I got a chance to talk to you guys. I know this is the beginning of a long and fruitful investigation by putting together engineering with biology concepts. I'm just so excited to see what happens and what you guys do in the future. Thank you very much.
That was wonderful, don't you think? The organizers of our school here would like to present Dr. Sishan with a plaque in recognition of his extraordinary effort. And thank you very much.
Thank you very much.
So we're going to take a break now for
7 minutes.
7 minutes.
Well, you could blame Scott for that.
Come punch me out later, I understand.
Is coffee outside?
always thought about installing Linux, but like
I never had a reason for it until a year after doing physics. We use Linux all the time.
Is it because like the programs you're running use Linux or?
Not necessarily. Well, one program. It is true. But it's really more so because you can do this SSH thing where you can connect to a remote computer from anywhere you have internet access. Work.
Let me answer it. Okay. Let's see here. Right now it's 73. 74 Fahrenheit. I think it is one of the but yeah, if you feel like should I give him a jacket? Okay, see if this is good enough or not. So, take some coffee. When you have coffee, this would have an hour roughly. Yeah, so you would have a, and then like leave a little time for reflecting. So, yeah, so that's fine. And then the next guy, yeah, you make it.
You're signing a contract for your house, or is it just like
Not yet.
but you're gonna have to?
I mean, it's a friend, right? So she's, you know, I trust that she's not going to
So you enjoying this?
Oh, you're renting from a friend?
Yeah. Mm-hmm. Yeah. Well, just, working in this position for a year and a half. I know where I trust her. I mean, once she can find out her schedule, her lawyer.
I'm wondering how formal I'm gonna
Just to see how compact it is?
Good question. Have you been to the tenant user?
No.
They might be able to help you with that.
Oh, it's not a contract. Like,
Yeah.
It's just like, is it more like it's too much? Because I mean, it's like.
Yeah. Oh, yeah. I mean, I don't know. I would go by the tenant union. Usually, what they do is they sort of serve as an advocate for students who are representing. You know, how is this going to work? They have a huge.
So you need to buy out.
So they can say, oh, your landlord has like 6,000 complaints. They're really bad. It's not up to them, or your landlord has no complaints. So they have to go. But they might be willing to.
Too? Yeah.
If
You're all loaded up, ready to go?
Student going to the they might be able to, they might be willing to sign it. It's like, it's kind of like student legal.
Going fast. Yeah.
You pay like a
Wait for the fast one.
tuition that gives you access to those services.
I don't anticipate anything.
Well,
上网去查刚刚。因为本来他的实验室网址，可能好像是新老师那个，找不到他的。
从你来的。
那我不知道，他就那个像大部分就是写上就是两极化，大部分都很喜欢他，有人说他会上错。
I don't know. His office is
Oh, he's not in the.
well, he's there. I think he's there, but he used to have the office like right first, like right on the right when you walk in the door, and now it's somebody else. So I don't know if that means that he's like transitioning. They just moved offices for some other reason. Or, yeah,
Yeah, because it seems like he's pretty competent, or he's really competent. Yeah, I did that. Well, do you have a primary care?
No, I mean, usually
Oh, just going to be kids.
but I graduated in December.
But do you actually have health insurance right now?
And so I do. But I just recently, like, I'm like, talking to Steve she's like, oh, they'll do your TV.
Oh, you hear your TV test?
I don't like
Yeah, it's just like a little shot.
So I went over there and then I talked to the business officer. She ever messaged me. Sucks after that.
我今天要找个借口说我要离开他，我有工作，我说我找到工作。
The insurance they give us,
我就随便找，
like grad school,
然后那个
it was like a little bit of dental. Yeah. I don't think I could have health insurance right now because I'm on my parents' plan. My health
Get in trouble, I know.
Is Hermes still running? Because I was looking at it.
You know, it is. I'm not sure.
睡得好吗？我问你，哎，想同样的事情，我回去就睡了。
I know some people are
大概4到5个小时，差不多。回去该上网我回去上网，写了发邮件，然后一下子就快来了。今天早上好不容易太感谢谢你，哎，真的太感谢，太感谢，不客气了，太感谢，因为我其实我在这儿我也有私心的，我也晚上很少睡。
Whenever I do something important,
带的是吗？喝料是带的好，是你带的好。我昨天抽东西，大家回去在讲，是吧？太棒了！哦，以后不知道再有没有机会再跟你一起，还有时间我们就有机会再跟你一起。啊，今天晚上干嘛？今晚休息，然后明天去 pub 去 pub。我还没去过，没去过，我真没去过。现在得可以去 pub。他这个做这个我问上他，做 structure， yes。
Okay, we're going to get started again. So next this morning we have Professor John Rogers from our Material Science Engineering Department here at the University of Illinois. John's been doing really spectacular, interesting technologies that he's going to talk about. So you're going to see this broad spectrum of some of the most wonderful things that you'll see for the interface between technology and biology.
Okay, all right, thanks for that introduction. It's a pleasure to be here and have a chance to talk to you about some of the work that we've been doing in my lab over the last 3, 4, 5 years or so. And it's pretty heavy on the engineering component, but we're very much interested in the interface to biology as well, as you see. as I make my way through the slides here. And it's an area of technology, I guess, that we're referring to as bio-integrated electronics, in the sense that the electronics is intimately in contact and able to communicate electrically, chemically, thermally with the tissue of a living system. And so I'll start by giving a motivation of what this field means to us. You know, it's a little bit of a perspective on how it's fitting with you know, other future directions in electronics and aligned with those efforts, actually, complementary to them. And then I'll switch over a little bit and talk about some materials and mechanics aspects of how to make electronics into a form that can accomplish this level of biointegration. And then conclude with some functional system level devices that we've built that appear to have some clinical utility for advanced surgical procedures. So if you think about electronics, and just as a side note, I mean, a lot of the slides that I'll go through here are pretty basic. I mean, if you're interested in a deeper level of detail, just let me know. This is very much high level, just to give you a sense of what we're doing and what the trajectory is. So this slide just provides kind of a primitive view of where electronics has been in the past, where it is now, and where it might be going in the future. So if you think about, half a decade ago or 30 years ago, this is the kind of computing systems that one would typically encounter. Large boxes would sit on your desk or fill entire rooms, and really by virtue of Moore's law scaling, transistors getting smaller and smaller, and the ability to pack more and more of them into smaller and smaller areas have enabled this kind of functionality to be reproduced in devices that, instead of having primarily industrial utility, are now valuable at the personal level because of the small size and the advanced functionality. And so if you think about where is electronics then going in the future, one dominant trend would obviously be to simply continue this kind of Moore's law scaling to enable even smaller and smaller devices, even more and more powerful, power-efficient computing systems. And that's certainly an interesting direction to go, and that will continue for some decades or so. The other area is a little bit less familiar, would be to focus on advanced display systems or components of these computing devices to make them larger in area, or maybe mechanically flexible, or offering other kinds of functionality. And that is a direction in macroelectronics that have different scaling metrics in terms of measuring progress, but is nevertheless an existing and continuing direction for electronics. The third thing that you might think about, I think about others as well, would be to move these things from the desktop to the individual, and ultimately integrate them in an intimate way, either on the surface of the body through a skin interface, or implanted into the body itself. And so these kinds of notions have been kicking around for 30, 40 years or so. So it's not a new field by any means, but I think there are new opportunities that emerge from new developments in form factors, shapes, properties, mechanical characteristics of electronics that allow one to come back to this kind of vision and maybe do some useful things. So if you think about existing bio-integrated devices, there are lots of examples. Again, this is an area that people have been thinking about for a long time. The ones that are in widespread commercial use are kind of summarized here, but I'm going to step through all of these examples, but just highlight a few. There are deep brain simulators that are used to deal with Parkinson's disease that involve electrodes penetrating down into the depth of the brain. Those are in widespread use. Cochlear implants to restore hearing to the hearing impaired. Other things, pacemakers, other kinds of devices, all of which offer some active level functionality. There are other kinds of devices that provide a more passive role, such as contact lenses. And if you look then at the range of stuff that's available, all of the active devices have this kind of construction, more or less. That is to say, it consists of two parts. One is the active electronics piece, which is typically in a rigid box. And then the interface to the biology happens through a small number of individual electrodes that penetrate into the tissue to form the interface. And that's fine for certain of these kinds of applications, but you can imagine that that kind of design approach is highly limited in terms of what you might be able to do, and also in terms of the long-term viability and biocompatibility of a system like this. On the other hand, some of the passive devices maybe embody a much more appealing design, the contact lens being an example of that, where the device itself takes on the shape, the form, the mechanical properties of the tissue that it's integrated with. And so contact lens is curved to match the curvature of the eye, and it's soft and water permeable to provide a much more robust and attractive type of interface to the tissue. And so, you know, the question that we've been exploring is to figure out whether it's possible to embed, you know, full active functionality that's available in something like a brain stimulator. It may be more advanced than this, but in a mode that integrates with the body much more like a contact lens might do. And so there are lots of potential contexts for utility of that kind of envisioned system. Probably the most obvious one would be to form an interface to the brain, either to provide kind of advanced diagnostic or therapeutic function for surgical procedure, or ultimately to provide an interface to a machine or prosthetic or something that sort. And so if you think about the brain, I mean, it's an electrical device, and if you wanted to interface to it, you know, a man-made piece of electronics, you might ideally want to bring to bear on that problem the full integrated power of silicon CMOS technology. But that kind of technology only exists in this kind of form. And so you can imagine that establishing an interface between these two things would be challenging because the shapes are totally different. And the mechanical properties are too. So the silicon has a modulus of about 100 gigapascals. The tissue typically will have a modulus between 1 and 10 kilopascals. So many orders of magnitude separation, and then the obvious challenge in geometry. And so what do people do? Well, this is the current state-of-the-art technology that people use to interface electronics. So the brain's been around for about 15 years. It's known as the Utah arrays, and I don't mean to denigrate it at all because it's been a spectacularly valuable type of tool for doing all kinds of research in brain function, but the mode of operation has certain kind of obvious limitations here. So the system consists of an array of micro machine sharp pins of silicon that terminate at their tips in an exposed electrode that then interfaces to the brain tissue. On the other side of those pins is a flat platform on which you can mount a chip that you might dice out of a larger wafer. And so you integrate those two things, pins and chips, and then use an air hammer to plunge it into the tissue of the brain. The pins penetrate and sort of accommodate the curvature, the mismatch in shape, the curved surface, reticulated curved surface of the brain, and the flat silicon chip. And that works okay for typically a few months, but you can imagine that's not a viable system in a long-term sense for two reasons. One is that there's a lot of damage to the tissue upon insertion, and then even after that, these tips tend to rattle around in the brain as the animal or the patient moves around. Because that brain actually moves in the head to some significant, in the skull to some significant amount, and then the modulus mismatch between these sharp high modulus pins and the tissue causes continued irritation that degrades the signal level here and also continues to damage the tissue. And so the vision then is pretty simple, which would be to try to form electronics that have you know, performance characteristics that we all know and love, you know, from silicon wafer-based CMOS, but in a form that's soft and shape conformal to the tissue to enable, you know, an intimate type of biointegration and long-term viability in the sense of being biocompatible. Okay, so that's the idea. That would be an example of what you might think of it as an indwelling biointegrated device. It's mounted on internal tissues, and I'll come back and describe some specific examples of brain-integrated electronics a little bit later in the talk. Other areas that we've worked on is in cardiac monitoring. The other point of integration, though, you could conceive of is on the surface of the skin. There would be a lot of interesting things that one could do there as well. The challenges are sort of similar to the ones I just described in the sense that the skin has tissue-like modulus that's much, much lower than silicon. It's also curved and sort of stretchy at some level. And so if you want to integrate electronics on the skin, you need to match all of those kind of properties in order to do it in a viable way. The other problem and challenge with the skin is it's constantly reconstructing. So it involves several different layers here. Cells in this base layer differentiate and migrate to the surface of the skin over time, where they eventually die and form this hard horny layer, sort of protective coating on the skin known as the stratum corneum. And this happens on a time scale of a couple weeks. And so if you want your electronics to sit on the skin, you need to worry about the modulus and the shape and whatnot. You also have to worry about this cellular escalator, essentially, and what you're going to do with the need to exfoliate these dead cells. And that creates a whole other set of challenges that we haven't directly addressed. We just highlight that as maybe an area of future opportunity with electronics that can kind of accommodate that kind of exfoliation and evolution of the underlying tissue. So in this area, as in the brain interface electronics area, there's been a lot of work over the years, and there are a lot of current research efforts. Last year we did a study for DARPA to sort of map out what the landscape of current work looks like. And these are representative of two classes of devices that we could sort of bin all of the existing efforts into in very rough sense. One is this sort of deep brain stimulator type of geometry where you have a brick of electronics, in this case, mounted on the body through straps, and then small numbers of electrodes interface with the skin. That's one direction that people are pursuing with its obvious limitations. The other is a little bit newer, but also a little bit primitive, which is to just glue chips onto tapes and then tape them against the skin. So the point I think here is that there are existing technologies. They have advantages and disadvantages. Maybe a new way of thinking about things could push the field forward. Okay. So if you think about then those kind of indwelling and non-dwelling electronic systems, the challenges really are in the interface. How do you do it? Long-term viability, but you still need optical electronic chemical access. How do you make things stick without irritation? Mechanics and shape are important here because they affect these two things. They also determine sort of wearability. And then there's a whole host of sort of DARPA-related metrics that would be important, for ultimate commercialization of these kinds of systems. So, cost, power, weight, size, this kind of thing. So what we've been trying to do is to realize a vision that looks like this in terms of electronics, which is to make electronics themselves skin or tissue-like as much as possible. And so what does that mean? That means making the electronics ultra-thin. That should be 5 microns. So much thinner than the skin, for example, much thinner than the surface layers on many internal tissues. Ultra light to minimize the mass loading, ultra low in modulus to minimize the mechanics loading. So that means circuits with moduli in 5 kilopascal range or smaller. If you could go smaller, that'd be even better. This is sort of the target we put up for ourselves. Shape conformal and perhaps air and water permeable and waterproof. It has to operate when completely immersed in biofluid. So the vision then becomes one of kind of an electronic temporary tattoo. So you're probably familiar with these kids' tattoos. They're very thin. They have many of these kinds of characteristics. And the interesting thing about them is once they go on, then the user loses all consciousness of the device actually being there in the sense that it moves freely with the underlying tissue. And so the question is, can you make silicon electronics into that form and design it to provide some kind of useful function? And that's what we've tried to do. So if you think about that as an outcome, it's useful to consider the backdrop of a sort of researchy but technology that's beginning to near commercialization, and that is organic electronics and flexible devices. So this is an area that we've been interested in for a long time. This is some early work from a team effort that we had when I was back at that allows you to team up with e-ink, which is a company now that provides the ink material for the Kindle, but this was the early days. This was 2000, 1999 or so. But then one interesting thing about that kind of material is it's thin and flexible, has the look and feel of a sheet of paper. And when you display, you need more than that, though. You need also a piece of electronics to turn on and off the pixels in that material to bring up images or text. And so we, at that time, were interested in organic-based electronic systems, so polymers or small molecules that could provide a semiconducting function in transistors that you can then form a sheet of plastic. And in this case, we built an array of such thin film transistors. We integrated with e-inks, electronic ink. You can make sort of a paper-like display. At that time, we thought that that might be an interesting type of consumer electronic device. And a lot of companies since then have pushed it forward and just developed the engineering sophistication, pointing out that you can really envision things like electronic newspapers that have the look and feel of paper, and that's moving forward. Now, this is not, in my opinion, a viable technology foundation for the kinds of biointegrated electronic systems I described before, for several reasons. One is that the performance that's available from these classes of materials is pretty lousy. It turns out to be good enough for that kind of display. If you want to do any kind of high-frequency electronics, it's not going to get you there. The other, and sort of reliability, robustness questions still remain, even though things are moving towards commercialization. The other limitation here is that in this particular form, the electronics are in a sheet of plastic, and so that's bendable. But in order to integrate with biology, you need something that can not only bend, but also stretch and flex and twist in ways that enable this kind of modulus matching. And this ain't it. So this is relatively still high modulus. I mean, the plastic is a few gigapascals, so still many orders of magnitude higher than the tissue. It's flexible. That's great for flexible display, but not good for biointegrated electronics, which is our present focus. So how do you do this then? So you like to do it in silicon. It's established material. People know how to build high-quality devices and circuits out of it. But you think of silicon typically in the form of a semiconductor wafer, which is certainly not bendable, low modulus, and not able to graft curved surfaces. So I'll just describe two very simple ideas that allow you to bypass that apparent limitation to build the kinds of systems that I just described. And the first idea is based on elementary bending mechanics, which dictates that for a given bend radius, the maximum strain in the material decreases linearly as you decrease the thickness. That's it. So 2x4 is not flexible. The sheet of paper is the same material essentially, but just by virtue of the fact that the paper is thin, it's bendable. So the same elementary mechanics applies to silicon. So if you can take a bulk wafer, which might be 0.5 millimeter thick, and slice it very, very thin, then the silicon becomes flexible. And so there are ways to do that. You can take a commodity 111 silicon wafer. If you do a two-step etching process, which involves a vertical trench etch, followed by top surface sidewall passivation and an anisotropic undercut etch with wet etchant like KOH, then you can slice off of the near surface region of the wafer very thin ribbons, wires, platelets, membranes of semiconductor grade silicon. And that's what's being shown here. So this is a cross-sectional SEM view after this undercut etching is proceeded for a little while, but not completely undercut the ribbon, just to illustrate what's going on here. Typically, in a real process, we completely undercut. And in that kind of thickness range, 150 nanometers or so in this particular case, the silicon becomes quite floppy and bendable, just due to that elementary bending mechanics that I mentioned before. So if you take that form of silicon, there are different ways to manipulate it and integrate it onto a thin, flexible substrate to make a flexible but still high-performance form of electronics as an alternative to this organic approach that we worked on and others are still working on over the years. So this is an example of an extreme implementation of that particular idea in the form of simple silicon CMOS circuit elements. So what this consists of are very thin ribbons of P and N-type silicon, and then other components, layers of material to form simple logic gates. So these are CMOS inverters, an array of them. And they're built actually on a very, very thin sheet of plastic. This is polyimide, just to illustrate how bendable you can make a silicon circuit. So in this case, making the silicon thin and also making the substrate thin makes this thing very bendable by virtue of the mechanics I just described. But you can actually do even a little bit better than that if you think a little bit more carefully about the mechanics. And so this also exploits not only ultra-thin plastic substrate, but a design that places what's called the neutral mechanical plane coincident with the most brittle material in these circuits, which happens to be the silicon. So the neutral mechanical plane is also a really simple idea. So if you have a sheet of plastic, let's say, just a uniform sheet of plastic, and you bend it like this, the top surface is in tension, the bottom surface is in compression, and the geometric midpoint sees zero strength. That's known as the neutral plane. And such a plane exists not only in a uniform sheet of material like I just described, but even in a multi-layer stack like this. It doesn't happen to lie at the geometric midpoint, but you can go off and calculate where it is. And in this case, we chose the thickness of the plastic substrate to put that neutral mechanical plane aligned with the silicon. So the neutral plane is indicated by the red dashed line here. And so that further enhances the flexibility of these devices to the point that you can, for example, wrap these circuits around the sharp edge of a microscope cover slip. So that radius of curvature is between 25 and 50 microns, for example. You can also wrap it around a capillary tube, fairly tight radius of curvature there. So I think the message here is that these simple ideas, you can make silicon circuits as flexible as you want them. I mean, down to a level that's almost foldable in some ways. But as I mentioned, in the context of that flexible display, flexible is not good enough. You need more than just flexibility if you want to do biointegration, because although you can wrap a cylinder or a cone, you can wrap a sharp edge, you can't even wrap a sphere, certainly can't do a body part or a complex curvilinear shape like the surface of the brain. So that brings me to the second simple idea, which is that if you make silicon thin and you also structure it into an accordion type geometry, then you can achieve sort of an end-to-end stretchability in silicon without inducing any significant strains in the material itself. And you can do that in a variety of ways. In this case, this is an image of that kind of wavy ultra-thin silicon bonded to an underlying rubber substrate. So the way this works, you actually stretch the rubber substrate out a little bit. Then you deliver to its surface these thin strips of silicon. It bonds through interface chemistry that you define explicitly. And then when you allow the rubber to snap back to its original dimension, it puts these ribbons into a state of compressive stress. And the way that the material responds to that compressive stress is through a nonlinear buckling instability that spontaneously yields this kind of wavy silicon. And so at a very intuitive level, the response of these wavy ribbons to strains that you apply, let's say along this direction, is very much compatible to your intuition of how an accordion works. So if you stretch this material in this direction, the amplitude goes down, the wavelength goes up in a way that avoids any kind of significant strains in the silicon. And the reverse thing happens if you compress it. The amplitudes go up, the wavelengths go down. Okay, so that's the intuition. Of course, the detailed mechanics are a lot more complicated than that. And because mechanics is playing a central role in the design layouts in these cases, we work with theoretical mechanicians to be sure that we understand all the fine details so we can optimize our circuits using this kind of idea. So that's the idea in its simplest form. That's not a circuit technology, obviously. It's just a pieces of silicon on a rubber substrate. But you can combine those two things. So these ultra-thin plastic-supported silicon circuits with this neutral mechanical plane design, combined with this idea of buckling and rubber substrates to achieve full integrated circuits that are stretchable. And this is an example of one of those kinds of wavy circuits. Again, this is one of these CMOS logic gates where we have a P-channel MOSFET here, an N-channel MOSFET over here, and then we have the wiring that connects them up in an appropriate way to make this inverter. And what you're seeing here is this circuit is supported on a thin sheet of polyimide like I showed you before, and then the underlying substrate is a silicone elastomer. And this is what it looks like after that buckling instability is invoked as a result of relaxing the pre-strain that's applied during the bonding step. So, again, finite element modeling and mechanics is central here. So this is an example of full 3D modeling of this particular structure. Now, what you see is the pattern of waves is actually a lot more complicated than in the case of just these uniform strips of silicon. But that's easy to understand because this circuit is both heterogeneous in materials. Silicon is here, for example, it's not over here, and also in thickness. So the local bending rigidity of the circuit varies depending on where you are in the circuit. So in this particular case, bending rigidity is lowest at these interconnect lines, highest here. So the waves tend to localize in those regions that are more easily bent than the locations of the silicon transistor. So this is just a short movie to show that buckling process, looking at top-down view through an optical microscope. There are the transistors. You can see this buckling set in as biaxial pre-strains relax in the substrate. So that's the physics at the level of its single inverter, but you can apply that idea chip scale. So here's a little bit more advanced system consisting of 10, these are 10 CMOS ring oscillators, just 3-stage ring oscillators. Here are inverters down here and individual transistors up here. So this is supported on rubber substrate. You can see the wrinkles and the wavy nature of this ultra-thin circuit. The photographer who took this is actually illuminating the circuit to highlight that kind of out-of-plane deformation due to the illumination angle. So what we're seeing here is the troughs appear black, and the raised peaks in these wave structures appear bright, just due to the way the light is reflecting. So it makes it look as though this thing has been deformed at a level that's a little bit more extreme than it actually is. So for these waves, the wavelength is maybe a few hundred microns, and the wave amplitude is a few microns. But that kind of mild wavy shape is enough that allows you to take this circuit and then stretch it back and forth this way or that way, or whatever way you want, without changing significantly the properties of the devices, or certainly not fracturing the silicon. To some limit, I'll come back to that in a second. Yeah?
Do you have waving happen in both?
Yeah, so you can see sort of wave structures here, and then also wave structures there, and they're following along.
To see how good is the react. Do you verify how is it fracture after a few cycles of this kind of buckling process?
Yeah, so that's a good question. So we have mechanical fatigue testing setups that allow us to cycle these devices back and forth. So we'll, in an aggressive test, we'll run up to maybe 100,000 stretching cycles up to some predefined strain level. We also do bending fatigue as well. So I think at an academic level, that's pretty good. For a real commercial product, you may want to go even a little bit beyond that. But these systems are designed up front with full quantitative modeling of the mechanics, such that we know what the peak strains and stresses are at different locations in the devices and the circuits. And so they're designed specifically to accommodate a certain range of stretchability. And then we verify that through this fatigue analysis. Somebody else have a question?
Do you have blockage?
So you can. So there depends on the thickness of the substrate. There is some residual stress in the system. So the top surface is a little more highly stressed than the bottom surface because it's mechanically loaded by the circuit. So if the thickness of the bottom substrate is sufficiently large, then you can get an overall bowing effect rather than local wavy shapes. And so I'll show you in a couple of slides a more advanced design that we more routinely use these days that avoids that kind of problem. Those are very good point. Yeah?
What's the purpose of using that polyimide substrate? Is it for having more flexibility in the structure?
It just provides, so actually
Can you repeat the question?
Yeah, so the question is why use the polyimide? Why not just the silicone, I guess is the question. So the silicone is a low modulus material. It's a modulus about 1 megapascal. It's slightly gas permeable. It's also permeable to certain classes of solvents that you might want to use in a semiconductor fabrication sequence. And so it's useful to separate the elastomer from everything associated with fabrication of the circuit. It also has a very large CTE. So we use the polyimide as a support for fabricating the circuits, and then use that in a final step for integration with the silicone. So polyimide is just a much easier material to work with during circuit fab.
Does it have good adhesion with the silicone?
Yeah, so polyimide is used in packaging applications currently in the silicon industry, and in some cases it's used as an interlayer dielectric. So in that case and others, we're usually trying to select polymer materials that have already had some validated range of applications in existing conventional silicon ICs. And so those kinds of problems have been worked out for us, and we try to just build on that and exploit it. Yeah. Any questions?
Is there any limitation about the circuit different way we say?
Yeah, I'll get to that. There are limitations. So this is a concept, but it's not magic, you know. So there are limitations. I'll come to that in just a couple slides. All right, any other questions or comments? Yeah.
How to control the fatigue loading about the stretchable silicon?
How to control
the fatigue loading?
So we control the fatigue by designing the layouts to avoid any stress concentrations at interfaces or in critical material elements. So it's really mechanics design that's done alongside of circuit design to create a layout that's robust to fatigue. That's one part of the answer. The other part of the answer is that monocrystalline silicon, in that sense, is good not only because it's attractive electronic properties, but it's also known to have very good fatigue resistance because there are no grain boundaries to migrate during cyclical stress loading. And that's already established from the MEMS community. And so, you know, we do our own fatigue testing, but I think there's ample reason to think that, you know, monocrystalline silicon is good not only for electronic properties, but also fatigue. Okay, so these circuits are not only stretchable, but they're soft, you know, at a mechanical level. And this was a nice image that people in our Beckman Institute took for us. We dropped off the circuit, and they came up with this nice posing, which I think is aesthetically appealing, but also kind of demonstrates the physics a little bit. So what this is one of these wavy circuits on rubber, and then into the center of this thing, the photographer plunged a glass pipette to induce some local deformation here. And what you can see is these wave structures move into form to accommodate this kind of local strain. So in this line back here, which is analogous to this line here, you can see that the wave structures are still present. In this particular case, though, the waves have sort of been pulled out as this line has extended in length to accommodate this local force. And so that kind of illustrates the physics. It also illustrates a limitation. So you can imagine if you continue to press this thing, this particular line is already flat. It doesn't have any geometrical ability to continue to stretch. And so it will fracture if pressed much beyond this particular level. So there's a finite range of stretchability in these systems that one can compute and then design around using approaches I'll describe in a second, or right now. So that's the idea. So there are two limitations here. One is range of stretchability. The other thing is that because this circuit is all interconnected together by that underlying sheet of polyimide, when this thing is pressed down, the silicon transistors see a bit of strain also. And the strain couples to mobility. So depending on how you're stretching or twisting or torquing this thing, the current outputs in individual devices could fluctuate up and down by maybe 20%, just due to that strain-induced change in the electronic properties. And depending on the circuit design, that may or may not be a problem. So you'd like to better strain isolate the active devices, those that are sensitive to strain, and you'd also like to expand the range of stretchability. So the particular range of stretching that a circuit like this can accommodate is maybe 20%. So that's maybe a factor of 10 or 20 larger than the intrinsic stretchability of the silicon, but it's not stretchable like a rubber band. Okay, so in some of these biointegrated applications, you'd want something that's more like latex, and this is not it. And so how can you bypass those limitations, but still retain silicon and some of the simpler ideas that allow you to get to that point? And the answer is that you do two things. One is instead of the circuit existing in a uniform sheet, you etch it into the geometry of an interconnected mesh. So this particular case is just a simple illustration of the idea. It's islands connected by straight bridges. So this is a thin sheet nanomembrane of silicon. And
structured into this mesh, number one. And then number two is by controlling the surface chemistry of the underlying silicone rubber, you can induce selective bonding of the silicon only to these island regions. So if you do the bonding of a mesh like that on a piece of silicone that's pre-stretched, and then you relax it, what happens is that the interconnecting ribbons deform out of the plane to form these kind of arc-shaped structures that accommodate all of the relaxation of pre-strain in a way that strain isolates these islands. So the concept then is to use this kind of idea, implement a circuit in which your active devices are in the islands, and then these non-coplanar bridge structures support the interconnect. Okay? So this is an example just in silicon. You can see with this kind of, those two ideas, you can really accommodate very large strains with a completely linear, elastic response of the overall system. So these things can stretch 100, 200%, much more like rubber band. So those are the ideas just implemented in a sheet of silicon. As I mentioned, you can use those concepts and extensions of them to build silicon electronics. And here's an example of that. So this is an island, a silicon CMOS here. And then these are, in this case, non-coplanar serpentine ribbon cables, essentially, that are connecting these individual islands together to form an overall system that has some kind of functionality. Now, the concepts of neutral mechanical plane are implemented in this kind of ribbon. So it's actually a polymer metal polymer stack where the metal's at the midpoint to improve the flexibility of that line. So you can do full finite element modeling. This is what these interconnects look immediately after fabrication. It's about 20% pre-strain, and this is after stretching to 60% or so. So the strain at the locations of the silicon device is very, very small with these kinds of designs. Now, in a real system, you encapsulate the top of this thing with another low modulus elastomer so that these fragile interconnects don't come into mechanical contact with the environment, which would obviously be a problem. So that's stretchable circuits. If they're stretchable, then they are also deformable in a way that allows you to wrap complex curvilinear shapes and also integrate silicon ICs onto a variety of unusual substrates. So here's silicon on paper. You can bend it and fold it without affecting the operation of the circuit. Here is silicon ICs on the tip of a latex glove using the ideas I just showed you. And here's an example where we wrapped silicon electronics onto the dimpled surface of a golf ball. And I have no idea what that's useful for, but you can do that just to illustrate the concept. So you can do it with silicon. Nothing I've told you, though, is particular to silicon. So any kind of inorganic, brittle semiconductor material can be used in this way. And so you do not only electronics, but photovoltaics, LEDs, photodetectors, anything you want. Here's an example of microscale LEDs, red LEDs based on Allen gap quantum wells that we grow on an epitaxial layer of aluminum arsenide, which can be selectively etched in HF to release these very thin active layers from an underlying wafer substrate. So the release process is a little bit different than what I showed you for silicon, but the concept is essentially the same. So the process for building these thin LEDs involves laterally defining their geometries by vertically etching down through the active materials to expose the aluminum arsenide. Then we pattern little pieces of photoresist that anchor these devices to the underlying layer after they're completely undercut etched by immersion in HF. So these devices are in a diving board type geometry anchored at their endpoints by these photoresist anchors such that they don't lose their lithographically defined locations and simply wash off into the etching bath after eliminating this aluminum arsenide layer. So this is what the wafer looks like. And then we have soft rubber stamps that allow us to come in and pick off selected devices from the growth substrate and then deliver them down onto plastic or rubber for making functional systems. So here is an array of such kind of ultra-thin LEDs interconnected using these non-coplanar serpentine ribbon cables I showed you before in the context of silicon electronics. So each one of those islands is an active LED, and you can see the interconnects capability to probe them here. The whole thing is sitting on a piece of silicone rubber. So you can make arrays of LEDs, very thin, very low effective modules. You can put them on a thin membrane of rubber, for example. You blow them up like a balloon without affecting the operation. So the system is designed to accommodate very large stresses and strains. In this case, sort of biaxial, but you can take these things and pull them down on the sharp tip of a pencil without affecting the operation as well. You can put them on rubber bands, twist them up, do all sorts of things to these devices without altering the operating characteristics. You can also integrate them onto a latex glove. This is an optical proximity sensor consisting of an array of these micro LEDs and then a corresponding array of tiny photodetectors on a latex glove designed in a mode that can be operated underwater. And I'll come back to that in a second because all these biointegrated devices need to operate under salt water, and that tends to be a challenging requirement, especially when you need to maintain these attractive mechanical characteristics. And I'll mention something about that a little bit later, but here's an example of this. We have a funded program with a surgeon who'd like to put smart devices on a surgical glove. This is the first example of that. Okay, so that's the mechanics, the materials, some aspects of the technology. At this point, I'll shift gears and talk to you about actual systems that we've built in collaboration with surgeons and clinicians to satisfy some needs for advanced diagnostic devices in cardiac therapy and in electrocorticography for diagnosing epilepsy. But before I shift gears, are there any other questions about the ideas or the approaches or what's happening here? Okay, so let me start with cardiac electrophysiology. So the clinicians that we work with are at the medical school at the University of Pennsylvania. They're cardiologists, practicing cardiologists, and one of the problems that they deal with are cardiac arrhythmias, so irregular beating of the heart. And typically that is induced by locally aberrant tissue, parts of the cardiac muscle that are not firing in a predictable and regular way. And the clinical procedure to treat that form of arrhythmia involves two steps. One is mapping the electrical activity on the surface of the heart or in the interior of the heart as the heart is beating to identify the location and spatial extent of the aberrant tissue. And they do that by electrical mapping. And it's typically done with a single point electrode that is moved from place to place on the heart while electrical signals are measured. And in that kind of point-by-point fashion, you can map out where the aberrant tissue is. And then the second step in the process is to remove that tissue. You either resect it or you ablate it to eliminate it and thereby cure the arrhythmia. So these kind of procedures, there's some mortality rate, and the mortality rate is most strongly linked to the time of the procedure, the overall time. And so the idea is if you could map the electrical activity on the surface of the beating heart much more quickly than is currently possible with this by-hand, point-by-point probing, you could eliminate a lot of the time associated with the procedure, and you could more accurately identify the spatial extent of the aberrant tissue, so you eliminate the minimum possible tissue in the ablation or the resection process. And so we built a device to do that kind of mapping that involves an array of electrodes that interface to the cardiac tissue. And each one of those electrodes at each unit cell incorporates a multiplexing silicon transistor and an amplifier. The signals are weak, and you need to be able to do active matrix addressing of this array of sensors so you can reduce the number of wires that need to come out in order to address all of the sensor elements. So this particular device has about 2,000 transistors, these silicon nano ribbon transistors. It's all a sheet of plastic. And then the outputs occur through an asymmetric conductive film that then goes to an external data acquisition system for collecting the data. So let me show you the operation of this device in what's called an epicardial mode. So this is an open heart procedure. This is a pig, it's not a human. So may still be a little bit squeamish. I certainly am, although I watched this movie enough times to sort of desensitized to it. But anyway, so pig models turns out to be a good model for human hearts about the same size for these Yorkshire pigs, which are about 90 pound animals. So this is the device. And so instead of the penetrating pins that I mentioned before, that brain interface, these devices don't involve any adhesives or any penetrating electrodes because the device itself is so floppy, the tissue is slightly wet. So you can almost imagine this being like a piece of saran wrap, a little thin plastic strip or sheet, just sticks there. And so you just laminate, it's very non-destructive mode for doing the interface. Of course, this is high-density silicon electronics. And this is the ACF film, comes out through a PCB board, and then there's a computer over here. So this thing mounts onto the heart as the diagnostic part of this cardiac ablation therapy, and then it can remain adhered as the heart is beating. So what I'm going to show you is this device operating in a purely diagnostic monitoring mode, where we can make these things multifunctional. So in this case, it's actually also pacing the heart in a sophisticated way. So you see the heart beat rate change dramatically. Somebody hit a computer key over here, and that changed the kind of pacing that was going on. We also have devices that can actually do the ablation. But what's published is only the diagnostic part of that process. So the thing is sitting on the heart is vigorously beating, but data is streaming out of the device at a very high rate, about a kilohertz per channel. So over the course of minutes, gigabytes of data is coming out, and it provides you a movie, essentially, of electrical potential on the surface of the beating heart that allows you to determine which tissue, region of the heart tissue is aberrant and which is behaving normally. So this is a movie of that kind of data collected from a healthy pig. So you're not going to see any aberrant tissue in this particular case. This is one spatial coordinate along one axis of the tape, and this is the other one. So this is position, and then each one of these pixels corresponds to the potential that's being recorded by that corresponding laminated electrode, rendered in a color scale in this way. And so this movie is actually dramatically slowed down compared to real time. This is an EKG trace, so take a single pixel and plot the potential as a function of time. This is one beat sequence. This is polarization, repolarization event, another polarization, repolarization. So this is the beat cycle. And the pig's heart is beating between 60 and 80 times per minute in these kinds of systems. So this is slowed down. This dashed line gives you a sense of where we are in the beat cycle. You see a wave of depolarization move across the heart. That's the way normal cardiac tissue coordinates contraction. And then you see a repolarization event there, preparing the cardiac muscle for another beat cycle. You see that happen again. Wave of depolarization move in that direction, and then a repolarization event here. So in terms of cardiac electrophysiology and mapping, this is providing unprecedented spatial and temporal resolution for the purpose of a surgical procedure. So this is not meant to be a long-term implant. Something sits on the heart during preparation for the surgery, and then it's peeled off and thrown away.
So how do you deliver this to the heart?
It's an open heart. Yeah. So the surgeon delivers it to the heart with forceps.
So then you find below.
So that's perfect lead into the next two slides. So there are two ways to do this ablation therapy. One is in this epicardial mode, which is on the outer surface of the heart. That's open heart. The other, which is gaining popularity because it's a less invasive procedure, is endocardial. So in that case, the mapping and the ablation happens via a catheter that runs up into the interior surface of the heart through an artery or vein. And so what we're trying to do now is take the electronic functionality that's present in that tape and implement it on a catheter balloon so that the balloon is inserted in the heart, is inflated, and then that pushes the electronics to the interior surface of the heart. You can do the mapping and the ablation that way. So we haven't published that. That's the direction we're going, and that's what this slide is showing. So in this case, you need stretchable electronics at a pretty extreme level. So the dimensional change in going from this uninflated state on this otherwise conventional catheter to this state is about 400% strength. It's huge. So you really have to be careful about mechanics design and how you're doing this. In this case, the ACF film, of course, doesn't have to be stretchable, but it wraps the length of the catheter and then terminates on one end of the electronics.
So when you inflate the balloon, it's not like on, right?
No, just inflates like that kind of balloon and pushes itself against the interior wall.
So your circuits will open up like flat.
They're opening up. They're moving with the balloon. So in this case, these are the electrodes, and they're following the contour of the balloon, whether it's in the uninflated state or fully deployed like that.
So it's the distinctive shape.
Well, the shape of that balloon, which is, yeah, has axisymmetric symmetry, not a simple symmetry. So that's what we're working on now, is that we put electronics on balloons, catheter balloons. But we have all these different building blocks. You actually make a very sophisticated, multifunctional catheter to do more sophisticated things. So here we have temperature sensors, LEDs, individual electrodes for stimulating, ablating, and measuring. And so we think there are a lot of opportunities in this kind of advanced catheter-based device for stretchable electronics. So that's the direction that we're moving. This is Marv Schleppian, who's involved in a lot of our work. He's probably the world's leading cardiologist specializing in cardiac ablation therapy. So he will do about 300 ablation procedures per year. It's almost one a day. And we work closely with him because he understands what are the limitations of existing technologies. So this example of Marv and some other collaborators, also another collaborator down here, if you can consider it that. So let me shift gears then and talk about neural applications. And here we work with experts in epilepsy at UPenn. And the problem is kind of similar. It's a lot more challenging, but it's similar to the one that I just described to you in cardiac ablation therapy, in the sense that in cases of patients who are suffering from acute epilepsy, what is done is the skull is opened up. These kind of coarse electrode structures, so each one of those is an individual electrode, is wrapped on the surface of the brain, and then while the patient is awake, a seizure is stimulated, usually by administering certain drugs. And the output of measurements made at each of these different electrode sites is used to develop a map of electrical activity on the surface of the brain. And then you try to identify the region of the brain that's responsible for the seizure. And then you just cut it out. That's the way it works. And so you can imagine this kind of technology is quite primitive. It's purely passive electrodes that are large and sparse. So the kind of spatial resolution that you can obtain is extremely limited. And you would like to, as locally as possible, define what region of the brain needs to be removed so you remove the minimum amount, obviously. And so the goal is to take a circuit system kind of similar to the one that we had for the heart and adapt it for the brain. The brain's way harder, though. You might not think it's as hard because the brain's not moving around as much as the heart. But the electrical signals are a factor of 1,000 smaller. So your amplifiers, your electronics have to be much higher in quality. And also the surface is a lot more curved. So the heart has a fairly large radius of curvature. It's otherwise pretty smooth. Here you have deep folds and trenches and all kinds of shapes that the circuit needs to conform to if you want a high fidelity, low impedance coupling between the electrodes and the tissue. So that's pretty hard. What we're doing at the moment is making just sheets of electrodes and then doing unusual things. So this is an example of active electrode sheet, similar to what I showed you before, but designed in kind of a foldable format. You can wrap it around a soft insert like this. You can actually plunge it into the gap between the right and left hemispheres of the brain. This is a cat model in this case, to study the function of the brain. But this does not solve, so this is interesting, we're pursuing that, but it does not solve the challenge of conformal integration on the surface. So you can do interesting things with flexible electronics, but you really want highly contoured types of systems for on-surface measurement. And so here's the problem. So this is a 2-scale model of a human brain. So if you make a thin circuit sheet like the one we use for the heart, you put it on the surface of the brain, the capillary action associated with the wet surfaces of the circuit in the brain are not sufficient to drive wrapping. And you can see that here. I mean, circuits are just too rigid to accommodate the sharp folds and surface of the brain. So that becomes a problem in mechanics that has a deceptively simple apparent solution, which is you can look at this system, you can ask when will a sheet of plastic wrap a cylinder with a radius of curvature given by R. And that just happens when the bending energy is lower than the adhesion energy. That is to say, this thing will bend more easily than the energy associated with the contact of these two wet surfaces. So the bending energy is cubic, and the thickness depends on the radius of curvature. The adhesion energy just depends on gamma, and in this case, this length dimension. So gamma is a given for two wet surfaces. So if you want wrapping, you want the bending rigidity as small as possible. You could imagine going to a low modulus material, but you have limited headroom there. You can drive the thickness down. The problem with driving the thickness down to the extreme levels that I showed you before is that the circuit then becomes too floppy to manipulate. The surgeon needs to manipulate it and mount it on the brain. So if it's so floppy enough to wrap the brain, it's not self-supporting. It just falls on top of itself. So for a 50 micron thick sheet of plastic, which is a good ballpark number for something that's thick enough to be mechanically manipulated, you can really only wrap a surface with a radius curvature down to about 1.5 centimeters, even when it's wet. You can't do smaller than that. And so that brings me then to a slightly different idea that we're pursuing, and I'll describe that and then wrap up. I just have a few more slides here, which is for indwelling biointegrated devices. There may be certain situations where you would like the electronics not only to integrate in the way that I mentioned before, but also resorb, just simply disappear over time. So you can imagine if you've done an operation and you want to provide some kind of electronics for monitoring the wound healing, it might be advantageous for that electronic to simply disappear into the body after the wound is healed, so you don't have to go back in and fish it out. And so the question is, can you come up with fully resorbable materials for electronics that would allow that kind of system? And there are some materials that appear to be attractive. So we work with Tufts University on silk. Turns out to be a very nice platform for electronics, and silk is fully biocompatible, bioresorbable. If you make electronics, in this case, it's partly non-resorbable, silicon devices here, and partly resorbable silk. So you can actually insert this into an animal. Over a course of months, the silk disappears, just leaving small pieces of silicon. So the idea is you could perhaps design this system, it's not fully bioresorbable, to eliminate any kind of adverse reaction by choosing the mass loading, the total amount of non-resorbable material to be as small as possible. So that's a concept. I'll describe in 1 second how that relates to this neural monitoring application. But the silk itself starts from raw silkworm cocoons. There are ways to purify this material and cast thin films of it that are robust platforms for the kinds of electronics I've talked about up to this point. And so this is silicon devices on silk. So it's flexible, it's bioresorbable, but at the same time it's high performance. So you get sort of wafer-like performance characteristics out of devices that are mounted on silk. And it's fully water soluble. So you can take a piece of this silicon on silk electronics and you drop it into water. The silk will quickly dissolve at a rate determined by how you do the processing, and then that leaves you just with silicon devices that just swim away. So one interesting question that we're actually pursuing is, in the context of these systems, is what is the minimum lethal dose of transistors? How many transistors? What is the size and shape that the body can accommodate in these kind of resorbable systems? Now, in the context of the electronics, what the silk platform allows you to do is make a system that's sufficiently rigid that you can mount it on the brain. You can mechanically manipulate it. Once it's mounted on the brain, you simply flush the entire thing with warm saline solution, thereby dissolving the silk and allowing an otherwise too thin circuit system to wrap onto the brain. And so that's what we've done. So here's an example of a silicon-supported 2.5 micron thick electrode array. Here is the ultimate embodiment, which is that thickness but cut into a mesh geometry. So it's originally supported by silk. Silk is washed away, thereby allowing this kind of conformal wrapping, in this case onto the tip of a glass pipette to illustrate what's possible. So you can do that on real animal models. This is a cat. So this is a conventional piece of flexible electronics. It does not wrap, as I mentioned before. This is an ultra-thin mesh of electrodes on silk after the silk has been washed away. So you can see this kind of conformal contact, even on this very, very curved, irregular surface of the tissue. And these are actually ECOG signals taken from the brain while the cat is exposed to a different series of images. And the green indicates a likely physiological response, and red is likely noise. So the much better contact yields a much higher fidelity monitoring. Okay, so that brings me to the end then. This is an area that we think is interesting. There are a lot of fundamental problems in materials, chemistry, and physics. I've mentioned a lot of these, understanding these interfaces and soft adhesion, micronanomechanics. But in the context of a set of engineering outcomes in surgical devices, implantables, brain-computer interfaces that we think could have larger societal value. And so we're pursuing those aspects as well. So just in conclusion, this is a list of the senior collaborators. It's mechanics, theorists, chemists, neurologists, cardiologists, biomaterials types, as well as EE growers like Shu-Ling Lee, who works with us on gallium arsenide epi materials. And of course, all the work is done by a really outstanding collection of students and postdocs in the group. And we just acknowledge them. Thank you for your attention. Be happy to answer any questions. Yeah. Well, it dissolves, and then the body internally metabolizes it. So it's known. It's an FDA-approved material for bioresorbable sutures. So it's a pretty strong material that can form into fibers, and it's commonly used to stitch up wounds. And in those cases, it simply dissolves and resorbs into the body. It's also edible. It's some organic material, so it's water soluble and also resorbable. Yeah.
How long does it take to dissolve the cell?
Yeah, so there are different ways to prepare the material that determines that time scale. So the movie that I showed you, is just a few seconds. But depending on the crystallinity of the material, which is determined by processing conditions, the dissolution times can be months. So that's kind of a tunable aspect of the material, set by the way the film is prepared at the outset.
So the film is like elastic, like
it's not elastic. It's just flexible. It's a bendable material. Yeah.
So we know the pacemaker and functioning fiber will control the heart rate. Yeah. And so, is any idea to have a material directly contact with the pacemaker, and we don't use that whole area to cover the cardiac?
Yeah, so pacemakers are typically, current ones are designed for point stimulation. It's not demanding for large arrays of independently addressed electrodes. But we do work with cardiologists at UPenn, Dave Callens, who is interested in more elaborate ways to pace the heart, in which you had a large array of electrodes, similar to the ones that we're using for sensing, but actually for the purpose of stimulating. So in that case, you would not have to pace the heart by a simple single point stimulation, but you could pattern the spatial temporal extent of the stimulation to do more sophisticated things in pacing. And so that's the direction that we're going, and we have devices that are, have been shipped out for animal testing. But I think that's a good point, and it's an area of opportunity.
And the other question is, do you any idea about the material, about liquid material?
The liquids?
Yeah.
Yeah, so I forgot to come back to that. So I mentioned it several times. I mean, in these kinds of devices, we first got started in this area probably 2.5 years ago. I would say that we spent a good 9 months trying to figure out how to make these things waterproof. And it was a problem that we didn't initially anticipate, because if you look at this kind of picture, you would say, okay, the tissue is a little bit moist, but it's not too bad. You can imagine a laminating circuit there and just running in that way. But whenever you open up a body cavity like this and you're doing some kind of procedure over time, you have to flush the entire tissue with saline solution periodically to prevent it from drying out. So these devices are completely immersed in saline solution, and associated biofluids during their operation. So the test for these devices then, before they're ever used in this way, is to completely immerse them in salt water, in a saltwater bath, and measure leakage currents. And if it's above about 10 microamps, then it's too high because it will end up shocking the animal. So the first attempt that we had at this was actually cat brain, and the device is laminated on, and as soon as it was flushed with saline, it completely catastrophically shocked the animal, burned up the tissue. It's like smoking. Smoke is coming out of the cat's head, with this thing, because we just didn't appreciate the problem. So the question is, how do you seal these circuits from salt water in a way that doesn't compromise the mechanics? So you can imagine you have one of these sheets, you could embed it in a block of polymer. That would do the trick, but then you don't have the kind of device you need. So we use all kinds of tricks like staggered via structures, multi-layer stacks of inorganics and organic films to form water barriers that are still sufficiently thin that the overall device is flexible. And so, like I said, it's probably nearly a year cycle time, all different kinds of recipes to achieve that outcome. And so we have ways to make these things waterproof, but it's a tricky thing. Yeah.
What kind of properties does the device measure?
This is just measuring surface potential associated with contraction of the cardiac muscle. Just measuring molecular potential.
So also this type of device, can you build out these things silicon sheets?
So this is actually the most sophisticated thing that we've done. Okay, so we're not anywhere near a microprocessor because there are lots of opportunities that don't require that. My own feeling is that the functionality could be quite sophisticated. I mean, there's nothing about these ideas that is feature size dependent. So you can imagine in a more realistic way to highly integrated systems, each one of those ribbons serving as a support for a large number of very small transistors, right? And then the islands are interconnected to one another, but each island is not just a transistor, may provide some level of processing functionality. So I think that would be the vision for how you go to much more sophisticated systems. But like I said, at this point, we're doing everything in an academic lab, and we're designing systems that satisfy a need as conveyed to us by our clinician collaborators. And those needs have not demanded anything more sophisticated than what we've done here. Now, having said that, if you look at the literature, a flexible integrated circuit that has 2,000 transistors is actually the most highly integrated flexible circuit that's ever been reported. Organic material, inorganic, independent of the details. So there have been flexible displays that have had more transistors, but it's a simpler circuit layout. So up to 100,000, let's say. But for an integrated circuit, 2,000 transistors is a lot, as benchmarked against the existing community of researchers who are kind of in this space.
So I'm just thinking that it would be possible that you probably have some additional devices, maybe building or living your body and do something like to do like long-term data collection.
Yeah. So I think that's a good point. So we are working on radio devices that would be able to serve as communication link. And we have the other question there is, what do you use for a power source? So you either make a battery, like a thin film battery, or what we're working on are inductive coils that allow you to power these things remotely. So those are good questions. Yeah.
Is it possible to measure a pace of the system?
Yeah, so we have multifunctional sheets that measure pace, and they also have the ability to do local ablation. So it diagnose, the whole thing would be over very quickly with very, very high resolution. So that's not yet demonstrated, but that's a multifunctional capability we're trying to do.
Measurement and ablation at the same time.
Right after one another, yeah. Measure and then ablate. So the surgeon really doesn't have to do anything by hand. You laminate the thing, you measure it, computer maybe does a pattern recognition, finds out where the ablation effort, and then does an RF ablation. So RF is localized delivery of RF energy is one of the predominant modes for doing the elimination of the tissue currently. Yeah, Jim.
Yeah,
You mentioned that the electronic property of these thin film will change with stretch. Is it mainly due to stretching or due to bending?
It's due to strain coupling to mobility. So anything that causes strain in silicon will change the mobilities of electrons and holes in opposite directions. So it's, I think it would be a difficult kind of variation to accommodate at the circuit level. So we try to avoid the issue by mechanics design. So the island non-coplanar bridge geometry really does keep the strain in the silicon to extremely low levels, even for large extensions.
But if you conform it to the brain, for example, there's large curvature.
Yeah, that's right. So that corresponds to stretching locally. So the island bridge design would be helpful there as well.
Yeah, when you're doing the conformal coverage of a convoluted surface like the brain, do you ever do something that would try to provide an overpressure, something that would get it to conform?
How to push it down? That's a good question. I mean, I think the approach that we're using with this dissolvable silk allows us to get into a regime of thickness and mesh layout that the thing is totally floppy. So it's really just water capillarity that's sucking it into contact. So you don't have to externally push it into contact. It just drapes itself down.
And the fact that that substrate disappears isn't ever a problem in terms of long-term measurements?
No, because it's fully resorbable. So it's dissolving away. It's in aqueous solution in the brain cavity. So it would be a big problem if it's not biocompatible and resorbable. So being water soluble is only one requirement. The other one is fully resorbable.
Okay, well, thank you very much for that very stimulating. The organizers wish to show their appreciation for your effort in presenting this report.
Great, thank you very much.
Yeah, thank you. All right. Okay, the last speaker this morning is a visiting lecturer from University of California, Berkeley. Daniel Fletcher is going to talk to us about mechanical regulation of the cytoskeleton. So some of the things that we've seen this morning, we've seen the biology, we've seen some technology, and soon we're going to see some things put together here and to determine function. So this will be quite interesting.
Great, thank you very much. It's a pleasure to be here this morning. So far today you've had two talks. One has been more basic biology, and the last one, fascinating discussion of technology and how it can be used to understand something about biology. My talk's going to fit somewhere in between those two. I'm going to tell you a little bit more about a specific biological system, in particular the cytoskeleton and how it's used for motility. And I'm going to try and talk about the ways in which the mechanics of the environment are important for the functioning of that biological system. So rather than the discussion you just heard about how mechanics or shape change is to be avoided, biological systems use that information. I want to show you a couple examples of where that is both good and where that is bad. I'm also going to talk to you about a couple different ways of gathering information, of sensing biological systems. In particular, I'll talk about optical methods and force microscopy methods for gathering information about how biological systems respond to their environment. So the picture I have up here is of a fish keratocyte cell. So if any of you have had a goldfish before, if you were to pluck a scale off of that goldfish with proper approval and culture that in a dish, off would crawl a sheet of cells composed of these individual cells. Eventually those individual cells would crawl away. And those are wound healing cells that participate in patching up the fish now that you've damaged the scale. And so these cells have been used as a model system for understanding many of the features of motility, of crawling motility. So I'll tell you a little bit more about crawling motility. What you're looking at inside of the cell is some fluorescently labeled proteins. So you heard this morning about actin. So in red is actin, and that's a set of polymers that are involved in pushing out the leading edge of this crawling cell. What you're looking at in green is a protein called vinculin, which is involved in attaching to the substrate. So the reason why this cell doesn't get washed away in fluid or on the outside of the cells, it's able to hold on. And then in blue is the nucleus. And for the kinds of things I'll be talking about today are the very short time scale processes. The nucleus is really kind of dead weight. Now the nucleus, of course, is important for the ability of the cell to maintain itself. But for many of the processes that I want to describe today, you don't need the nucleus. The nucleus is there as a reference, but all of the behaviors that I want to talk about really rely on what proteins are present and their ability to interact, sense, and respond on their own. And you can actually dramatically show that you don't need the nucleus by slicing it off of these cells, and the cells still crawl. So the questions that my lab is interested in are twofold. How do cells respond to their physical environment? What are the signals that are interpreted? How are they interpreted on the molecular scale? And what sort of responses do cells undergo? And then secondly, how can we rebuild these processes? Are there ways of generating both new understanding about how cells are organized and how they're built, as well as creating new sensors? Is it possible to take some of what's known about individual molecules, about how they're organized, put them back together, and use them in some engineering way? So I'll talk a little bit about the second, but mostly about the first. So the outline for today involves a few different things. First, I want to motivate why we're studying this process. Second, I'll tell you a little bit about some of the tools that we use for this, in particular force microscopy tools. And then I'll show you 3 different examples of where mechanics plays a role in biology. In the first case, leukemia, this is a situation in which mechanics is playing a negative role. There's a problem associated with leukemia that is due to changes in mechanics. Platelets, which are critical for a range of different repair mechanisms, I'll talk a little bit about how they are able to sense things about their physical environment. And finally, I'll go inside of the cell and talk about actin networks and how those isolated networks are able to respond to forces as well. And finally, I'll end with a discussion of some of the tools that we're working on to allow us to reassemble more complex systems. Okay, so let me start with some movies. My background was in engineering and applied physics, and when I was towards the end of my PhD working on developing microscopy techniques, I became enamored with motility, movements of cells. I think it's worth remembering, as you look at a swimming cell like this organism, that on this scale, so that disk is about the central part of the cells, maybe 6 to 8 microns in size. And there's no amount of engineering that we can do that can reproduce the kinds of motions that cells create. And so they really represent a remarkable engineering challenge, both for us to understand how they work, and then a challenge for fabrication. Can we actually put systems together, either out of biomolecules or other materials, that would let us get these sorts of behaviors? And so that's what really attracted me to movement, was the fact that here is an example of a microsystem that is able to do dramatic things, and we really don't understand how to do that. Granted, biology's had quite a bit of a head start on us, but still, we should be able to understand molecularly what's going on. Does anybody recognize this organism? You probably don't run across it on a daily basis, but if you've ever been hiking in the mountains and you're told not to drink the water because it might have something in it, that's sometimes what's in it. It's not in this form, it's in a cyst form, so this is Giardia lamblia. And if you feel really sick after going hiking, this may be the cause of it. So these beautiful organisms colonize your small intestine, and they're able to hold on. And actually, the mechanism of attachment is a subject of debate still. But they're able to swim around, and they're able to move thanks to these flagella that are flapping around. So in some parts of the body, infectious agents are able to swim. Here's another infectious agent. And this is called spiroplasma. Hopefully you haven't run across this one. This one really is nothing to fear. This is if you're a honeybee or a plant, you would be a citrus plant, you'd be very afraid of this. But other than that, these spiroplasma, these are molecules they don't infect humans, so there's nothing to be afraid of. But they have a remarkable ability to move. They have no external appendage. You saw that flapping tail on the Giardia. Those flagella are able to move the Giardia around. In the case of spiroplasma, there's no external appendage that allows it to move. And so in studying this organism, what we found was it has a helical shape, and what it does is it changes the helicity of its body, and it propagates that down in the form of a wave, and then starts at the head and switches the helicity of the body back, propagates it down. So it's able to swim through the viscous material without an external appendage. It essentially uses its body as an external appendage.
And how big is this?
So this is 3 or 4 microns long and about 200 nanometers wide. So a really, really small organism. One of the simplest ones out there. So here's an organism to be afraid of. Again, one that you can get infected by. If you go and have dinner and you have undercooked chicken or other products that haven't been heated high enough temperature, you could get Listeria monocytogenes. This is a particularly nasty bug, or if you have really soft cheeses as well. That's in France. It's expected that a certain number of people will die from Listeria monocytogenes because they don't want to have to pasteurize all the cheese. So this is a remarkable bacteria, and it also doesn't have an external appendage, but it's able to move. And it's able to evade the immune system so that your body doesn't even know it's there. And the way it does this is by getting inside of cells and then moving around as a result of its ability to hijack some of the host cell proteins and move. And in fact, the proteins that it hijacks, the major one, is active. So it's able to redo what cells are doing when they crawl. It's able to move itself around with that process. So question?
So what's the cytoskeletal element involved in spiroplasma movement? Is it
Good question. It's not clear yet. So there are a number of fibril proteins that are present, but exactly what the mechanism is that's causing this switch, we don't know. Good question. So Listeria monocytogenes, we do know, and there are a number of other bacteria that also hijack the host cell cytoplasm with proteins in it to be able to move around. So the kind of motility that I want to tell you about is not the swimming kinds. It's rather the crawling kind, because in most parts of the body, you don't really have a lot of space to swim, but there is space to crawl. And so let me show you what that fish keratocyte is able to do. So again, this is the cell that I showed you in the very first picture. And if you do observe it over time, so this is about a 5-minute movie, it's able to glide across a glass cover slip. So this is like it moving across a relatively flat scale as it tries to find the fish and patch it up. And it has this beautiful shape, and it's able to maintain its movement, move in a very highly directed way over long periods of time. Now all this process is taking place over 5 minutes. So what sort of thinking do you think this cell is doing? If thinking is even the right word. It has to process information about its environment. It has to know something about, am I going in the right way? Is this the best decision or not? So it's having to rely on chemical sensing. And what I want to show you here is physical sensing as well. So here's a slightly different situation for this same cell. So it's about to run and hit this barrier. So this is a little line at the top. It's really about a 5-micron step. And so it's crawling and doesn't have eyes, of course, so it doesn't know that this step is ahead of it. So how many people think that when it hits this step, it's just going to stop? So that's option number 1. Number 2 is that it's going to crawl right over the step. Doesn't care about a step. Number 3 is that it's going to bounce off like a ball hitting a wall. So how many people think it's just going to give up? Okay, good. So you have some faith in biology. You have some faith that cells will continue to process. How many people think it's going to crawl up and over?
How big is the cell?
So the cell is probably 20 microns across, and the height is under a micron. So you think people think it's going to cross over?
No.
And how many people think it's going to bounce off? All right. Okay. So let's see who's right. And if you're right, you can leave any time. So you already you can go to lunch early. So it's hitting, smash into it, and it bounced off. So bouncing off, people are right on. So in order for it to do that, it's had to completely reorganize over the course of about 5 minutes. It's the organization of its cytoskeleton, the internal structure that is allowing it to generate force, that's allowing it to hold on to the surface and allowing it to move. And you'll notice as soon as it hits, you begin to see some ruffling there at the edge. So the fact that it's hit one place is showing up in another place. So there is some sort of signal transduction, there's some sort of transfer of information about that contact to distal location.
So then,
yep.
If the barrier is of the same height as the cell, then, well, we know that if the barrier is very little, maybe a few nanometers, certainly it will
It'll go right over. Yep. Mm-hmm.
So
if it's closer to the height of the cell, actually the cell extension at the very leading edge is around 300 nanometers. It's quite thin. So if the height is around that, it'll go up and over. It's larger barriers like that. When it realizes I can't get around that, I'm just going to bounce off of it. Now, immune cells like neutrophils or macrophages don't have as flat a protrusion, and so they're able to go over much more complex geometry.
What's the barrier?
So this is just a step in a PDMS substrate. So the point of me showing you this was to point out that the very process of crawling has to have built into it some ability for the cell to sense, some ability for the cell to recognize that it's not going to be able to go forward, and it has to somehow reorganize. And the question that we wanted to tackle was, what exactly are the molecular mechanisms that allow for this sort of reorganization?
do I also see some deformation of the PDMS barrier?
This has been, there's a little bit of an overhang, I think, and so you see a little bit of the cell either going under or over that, but the bulk of the cell does not make it over the ledge. Okay, so I'll show you one more and perhaps the most dramatic example. Question?
Does the stiffness of the barrier affect whether or not it'll go over?
That's an interesting question. I expect that it would, because the mechanism that we think is at work is an ability for this cell to sense whether or not it can push. And so if it can push, then it's more likely to continue forward. But we haven't exhaustively tested that. So this last example is one that shows a coordinated movement. So this is a blood vessel of a fish. So this is a zebrafish, which is a model organism that many people use to study wound healing. And what you see at the top there is a puncture wound. So someone just took a needle and poked a hole into this zebrafish. And the question is, what is the body's response to that? What is this organism's response to that? And what you see on the lower part of that image is this blood vessel, and from this blood vessel come white blood cells. And so they are also crawling, and they're recognizing that there's a wound in that location, thanks to the presence of bacteria that have entered as that needle was poked into the fish. And so that chemical signal is being smelled by these cells, and these cells are moving towards that location. Now remember that these are about 10 micron in diameter bags, and they're filled with enough molecules and enough sense to be able to figure out which way to go, traverse a very complex physical and mechanical environment, and end up there. And they don't just sit there. Once they're there, they participate in the process of clearing the wound of invading bacteria and other organisms. So these are highly complex and highly capable structures. And the question is, how really do they do this? How do they traverse their mechanical microenvironment? Yeah?
So is this a section, or how exactly was this image got?
So this is not a section. This is the fin of the zebrafish is very, very thin. And so you can actually do light microscopy through it. You'll notice, for those of you who have done microscopy, you'll notice that there's a bit of a shadow. It kind of has that three-dimensional look. And that's characteristic of differential interference contrast microscopy. Yep.
And how long does it movie take?
I don't remember how long this is, but macrophages or neutrophils crawl something on the order of a micron per minute. So this is sped up maybe 20 to 30 times, something like that. Yep.
I still have one question for the cell. Do you have some coating of extracellular matrix or barrier?
No, you actually don't need, for these fish keratocytes, you don't need a specific extracellular matrix on that substrate. These are cells that are used to living on the outside of the cell, and so they don't depend on there being a certain ECM present. That's very different from most of the cells in our own body, which do require specific adhesions to specific ECM proteins. But that fish keratocyte doesn't. All right. So I want to focus in, and I'll be referring back to the actin cytoskeleton as I go through the examples of movements and mechanical interactions. And the actin cytoskeleton, I think, is a remarkable structure. It's kind of like Legos or Lincoln Logs. I guess those are quite old. In its ability to form a variety of different structures. You can find it in a huge range of different processes. I've talked about it involved in motility. But its ability to, a cell's ability to take up material through endocytosis or macropinocytosis, other processes for taking in material, all rely on actin. The same for getting rid of material or for releasing proteins, exocytosis. Movement of endosomes within cells also involve the assembly of these proteins. And more dramatic processes, like I was talking about for white blood cells, engulfing whole bacteria require this process. So these are kind of like the I-beams in this building. If you imagine this building was tired of being in the rain and wanted to move to, say, Berkeley, where it's nice and sunny right now, you could imagine disassembling all the I-beams and then somehow rebuilding them as the building moves towards the coast. That's kind of a complicated thing for us to do, but that's exactly what the cell is doing all the time. Its ability to translate from one location to the next means it has to take down its material in one place and reassemble it somewhere else. And that process of polymerization and depolymerization is central to the ability of actin to participate in these different processes. So actin, as I mentioned, forms different structures. I'm going to focus primarily on a branched actin network system. But it's worth noting that these same filaments can be organized in a range of different ways, from protruding bundles to stress fibers to cortical networks that are involved in cellular rigidity. Okay, so let's go back to this fish keratocyte cell and told you a little bit about what those proteins are. The F-actin that's involved, filamentous actin that's involved in generating these protrusions, the vinculin involved in holding on to the substrate, and the DNA that is along for the ride. Okay, so these dendritic actin networks are called dendritic because of the highly branched structure. So what you'll hopefully see here is that we have individual filaments, and off of filaments are formed daughter filaments. And there's a specific branching protein that's very important in this process called ARCP3. So this branching protein will continue to form new filaments, and there's another protein involved that will eventually stop the growth of these filaments. And so you have this process of growing new filaments, stopping that filament, another new filament grows off of it. But there's one key additional ingredient, and that is you can only bind this branching protein if you're close to a membrane. And so that's why all of these branched actin networks grow near membranes, whether it's the leading edge of the cell, or it's at the membrane as it's undergoing phagocytosis, or other membrane-associated processes, because turning on that branching protein requires you to be near a surface. So this is a clever way for cells, or for biology in general, to be able to pattern assembly of structures. Rather than just having a soup of proteins that are mixing together and assembling wherever, by restricting one particular step of that process to a two-dimensional surface, that leads to the highly oriented branch structure that generates a force in one direction. So we'll talk about that in more detail a little bit later.
Are they disassembling at the back end, as it were?
That's right, that's right. So there's an assembly process and a disassembly process, and there are different sets of proteins that are involved in the assembly as well as the disassembly. And maybe an important thing to point out is that there's a link between these growing networks, which are generating a force as they add subunits to the end, and the adhesion sites, these little green things. At some point within the network, within the cytoskeleton, these force-generating structures have to couple to the substrate, or else you're just going to push the back of the cell out. So there's got to be some link between what's being pushed out and what's being held in place. And so that's the coupling between the force generation and the adhesion. Okay, so let's talk a little bit about, I've been talking about forces and generating forces and resisting forces and compliance. How do you actually measure these forces at the molecular scale? So there are a number of different ways, and all of them essentially rely on springs. So if you understand how a spring works, you pretty much understand how these sorts of force microscopy tools work. So with a spring, if you are compressing that spring, you can figure out how much compressional force is being generated. If you're pulling on that spring, you can figure out how much tension is generated. But you need to measure something. And typically, you need to measure two things, basically. You need to know what that spring constant is, and you need to know how far the spring moved. And so the techniques that are used for force microscopy differ quite a bit on what is the spring, and how do you know how much it's moved. But in general, you're always thinking about those two things. What's my spring? How much is it moved? So two tools that we use in lab are one, optical tweezers, and two, atomic force microscopy. And I'll talk mostly today about atomic force microscopy because it has a number of applications in both cell biology as well as understanding molecular mechanics. In the case of optical tweezers, the spring is the potential, the gradient of the field created by focused light. And movement of a trapped particle within that field can be tracked by yet another field that allows you to quantify displacement on a very small length scale, a nanometer length scale. Now, the spring constants of these optical tweezers are quite small on the piconewton per nanometer scale. Atomic force microscopy has a range of different stiffnesses that are possible. Conventional cantilevers that are used or that are commercial are on the tens to hundreds of piconewtons per nanometer. So AFM. Now AFM, some of you may know, was really not developed for biological applications. It was developed for material science applications. The people who developed it wanted to be able to image the topography at the atomic scale, hence the name atomic, for non-conducting samples. So just before development of the AFM, the STM, the scanning tunneling microscope, was developed. And it relied on the fact that a tunneling current was passing between your probe and the surface you're interested in. And based on that tunneling current and a good feedback circuit, you can look at the topography of your sample. Now, that only works if your sample is conducting. So what if you want to look at a non-conducting sample? Well, that's where AFM came in. So AFM was originally just a conducting cantilever that had a sharp tip, and then an STM on top of it. So the STM idea was used to measure the distance, and then the spring was this little gold foil cantilever. And so now, with microfabrication tools, the cantilevers are now fabricated. They have tips with controlled sharpness, and rather than using an STM on top, we now bounce lasers off of them. So the current configuration that most people use for AFMs looks something like this. You've got a cantilever base that you can move in 3 dimensions. You've got a laser that's bouncing off of the end of your cantilever, and you've got some position-sensitive detector that allows you to sense what the displacement is, both how much it displaces vertically as well as laterally, if that's important for your measurement. Now, for biological applications, it was realized not long after development of AFM that you could immerse the system, and lasers will propagate just fine through most media, and as a result, you can do the same exact sorts of measurements to understand compliance of a material that you want to do in air, you could also do them in liquid. And so a standard configuration for measuring mechanical properties of individual cells looks something like this. You've got a cell in the media, you've got the same cantilever, and you're bouncing a laser through that chamber. The kinds of information you can gather from AFM measurements are the traditional topography, the height information. Where is my cantilever for a particular force set point? But if I do something known as tapping mode, I can look at other features. I can look at the amplitude of the tapping signal or the phase of the tapping signal, and I can get different information about the interaction with that surface. So you saw in the last talk, the information that you're gathering was a potential. It was an electrical potential from a surface. Here we're gathering very different information. Topography, stiffness, and I'll show you how much force is being generated by a particular cell. Now, the AFM by itself is a useful tool, but there's been a lot of interest in harnessing optical microscopy at the same time as force microscopy. So many of the AFMs that are now in use are in use as coupled instruments, where there's optical microscopy at the same time as AFM. And that simply means that while you're doing AFM, so the top is a phase image, and you can see fluorescent beads that are highlighted there. The bottom image shows some of the labeled proteins of those cells. You can combine the power of fluorescence microscopy, which allows specificity. You know which molecule you're looking at, and you can know how many there are, you can know where they move, etc. You can combine knowledge of the molecules with knowledge of the mechanics. So that's the motivation for combining AFM with optical microscopy. How many people have used AFMs here? A few people? Out? Okay, good. So there's some knowledge of AFM. All right, so perhaps the simplest experiment you can do with an AFM is to learn something about the stiffness of the material that you're looking at, or the cell that you're looking at, or about the adhesion properties of the cantilever with the surface, or if you've attached the cell to that cantilever, of the cell to another cell. So the ability to measure stiffness or measure adhesion forces simply involves moving your AFM up and down on a surface. And the surface of the cell is the sample of choice here. And what you're doing is you're saying, if I push and I displace my cantilever, I can measure that displacement, but I also know how much I moved down, so I know how much I've deformed my substrate or my cell. And based on that, if I go over a range of different displacements, I can fit that to a curve, and that curve fit, based on a certain type of theory, will get you an elastic property. So you can get a Young's modulus, essentially, out of a cell. Now, I should note a caveat here. I'm not a big fan of quantifying things in terms of Young's modulus, especially something as complex as a cell. Maybe an analogy is like if you were to crush your car, you could get a force displacement curve out of that. Here's how much force it took to compress my car. But it doesn't really tell you a lot about the car. And I feel like the same is true with some mechanical measurements of cells. Unless what you want to know is an overall bulk property, because that's important to the system, then single measurements on cells don't tell you a great deal. Now, I'm going to show you some single elasticity measurements on cells, because I think it is relevant for one of the examples I'll tell you about. But in general, I think it's worth thinking hard before doing measurements like this. Why is that data relevant? So this is the kind of traditional setup for doing AFM with cell biology. And I wanted to point out that there are a range of different ways that you can configure cantilevers in order to do more complex measurements. And so a few of the systems that we've developed in my lab involve using multiple cantilevers, or being able to work with non-adherent cells, or look at coupling. So let me tell you briefly what these are. The dual cantilever system uses two adjacent cantilevers to be able to normalize out for drift. Those of you who have worked with AFM know that drift is a real big pain. The cantilever base essentially moves relative to the substrate, and as a result, you don't really know where you are. You don't know what your reference point is for long timescale measurements. So to deal with that, we developed a system that uses one cantilever to figure out where you are, and a second cantilever to measure a sample. In cell biology measurements, you often want to know what's happening in the plane of movement. That is, if a cell is contracting against a force or protruding against the cantilever, you want to know what's happening in that plane. But most microscopes are set up to look at the bottom, which is not in the plane of force measurement. So we built a side view imaging system that lets us watch from the side as cells are contracting or protruding. In the case of non-adherent cells, pushing on a cell is like pushing on a grape. It'll slide out from underneath the cantilever. And so to confine that, a simple hole in a substrate, microfabricated, was enough to solve that problem. Many times you want multiple points. You want to know what's happening at multiple points on a cell surface. And so combining AFM, either two cantilevers or one cantilever and a fluorescence probe gives you information about propagation of stresses through a cell. Okay, so let me start with a couple of the examples I told you about. And I'll start with one that is related to leukemia. So as a background bit of information, hematopoietic stem cells give rise to all the blood cells that we have in our bodies. Most people are familiar with the red blood cells and maybe the white blood cells, which are all these ones on the left. But there are also the platelets as well as some immune cells that are involved in surveillance. So I'll be talking about problems that occur when the hematopoietic stem cells don't differentiate all the way to the end cells. So leukemia is simply a cancer of blood cell precursors. That is, leukemia occurs when the body doesn't generate enough of a desired cell. So if it realizes that you don't have enough neutrophils, it's going to keep cranking out the precursor cells for those neutrophils, but they don't divide properly, so you don't end up getting any neutrophils. You just get a bunch of these precursor cells that don't differentiate. And the same is true on the lymphoid side. So the result is this. So on the left is a normal, healthy blood smear. So if you take a drop of blood from your finger, you smear it on a slide, stain it and look at it under a microscope, you see mostly red blood cells, these little things, and then a few white blood cells. That's about the right number. On the right-hand side is a leukemia patient. And so you can see many, many more blue cells, which are the white blood cells, as opposed to the red blood cells. And that's simply because the cells are not differentiating properly. They're not creating the right white blood cells. So what's the impact of this? Well, one of the ways that patients can die from leukemia is a process called leukostasis. And so leukostasis is essentially clogging of blood vessels. So these blood vessels are about the right size. You can see that they'll fit maybe one or two or a couple blood cells through them. This should be the same size as these vessels, but it has gotten much bigger. And the reason it's gotten much bigger is because of clogging of this vessel. Now, what can give rise to clogging? There are a number of different ways that leukostasis could occur. And we wanted to explore the possibility that limited cell deformability was a problem. And so this is one example where measuring an average modulus or average elasticity for a cell might be a useful thing. If white blood cells have to pass through small vessels and they have to deform when they do that, then being able to deform is a very important part of that white blood cell. And if they can't deform, just as trying to push something through a small tube, if you can't deform, then you're more likely to get clogged. So we wanted to explore whether that was the case. If we look at leukemia cells versus healthy cells and look at their mechanical properties, is there a difference? What we found was, in fact, there was, if you look at patient samples that are symptomatic for leukostasis, the average stiffness is greater than those who have leukemia but do not have signs of leukostasis. And so you can look at the difference. There's quite a significant difference. And that's just taking average measurements of stiffness of these patient samples. Now, perhaps more interesting is how you treat leukemia and what effect that has on stiffness. So the standard way of treating leukemia is chemotherapy. So you give a drug that essentially kills white blood cells. Kills many cells, but in particular white blood cells. Now, what happened when we exposed myeloid cells, in particular, to a chemotherapeutic, daughterubicin in this particular case, we found that the stiffness of those cells shot up over time, while for control cells, nothing happened. So simply the fact of giving leukemia to patients causes a stiffening of those leukemic cells. And it progressed even before apoptotic markers, which would say, okay, this cell is dying, it's in the process of dying and going to be removed from the bloodstream. The stiffness increased. And so what's responsible for that stiffness increase? What we found was if we use a drug that disassembles the actin cytoskeleton, we're going back to who's the culprit here? What is responsible for this change in stiffness? If we look at the actin cytoskeleton, that it seems to be a major part of this stiffness increase. So if we disable the actin cytoskeleton, then the stiffness change is not as pronounced. And so what this suggests is that the stiffening of white blood cells during, both before and during treatment for leukemia may be a causative factor of clogging of the vessels, which can result in rupture and then death. And so this is one example of where mechanics plays a central role in potentially in a clinical disease. But AFMs are not convenient things to have in a hospital. You're not going to probably find any hospitals that routinely will measure samples with an AFM. So there's got to be a better way of quantifying stiffness. And so we set out to try at least one way, and microfluidics offers some possibilities. And so we simply built a system in which we could look at the transit time of different cell populations. And so using a very simple flow system with channels that were fabricated to emulate the smallest capillaries that these blood cells would go through, we found that we could quantify the time it took to transit across a small chamber, and we could use that time as a measure for the stiffness. So an example, if we took a normal neutrophil and we pass it through one of these chambers, that's the same neutrophil going through, not different neutrophils. You can see that it moves relatively quickly. Now compare that to a neutrophil that's been activated. And so it's been activated with the same chemical that caused the neutrophils to come out of the blood vessels to the puncture wound in the fish, FMLP, and that activates them, stiffens them, and you'll see the same channel is much slower to go through. And that simply has to do, there's no adhesion, there's no coating on this channel. It's simply flowing the cells through. You can see that there's a difference in the time scale. Now, time scale and stiffness correlate. They're not perfect, but they provide some indication of which cells may be most prone to aggregation and to leukostasis. And so here's an example of patient samples as well as some control samples, and transit time being much longer. So this issue is not just confined to leukemia. There are a number of other diseases in which blood cells or other cells are affected, and stiffness may be playing a role in transit. Okay, so I want to go to the second example, and it's also going to be a blood cell, but I want to show you a couple movies before that. And that has to do with forces generated by individual cells. So take platelets, for example. Platelets are able to generate forces during clots. And most cells in your body are actually able to generate forces, contractile forces, because they're not going anywhere. Only a few cells in the body are really motile. The rest are involved in a contraction. So I'm going to show you a movie of mammary epithelial cells. So these are epithelial cells that are charged with growing a branched network. And what you'll see is in this gel, there are a set of different particles. And the different particles are, you can see the cell is growing, but the particles are also being pulled inwards. And this ability to contract is also thanks to the cytoskeleton. So the cytoskeleton, combination of actin and a molecular motor called myosin, are able to pull on adhesions and therefore deform substrates. So here you're seeing a combination of motility as well as contraction of a growing tissue. So can we measure these kinds of contraction forces with existing force microscopy tools? The answer is yes. AFM and related techniques are able to quantify the scale of forces that these types of cells are able to generate. And the method for doing that is simply contraction between two surfaces. So just as the cells are pulling on different adhesion sites, the ability to mount the cell on one surface and a second surface and watch as the cells contract, we can quantify again with this spring. If we know the displacement, we can know the force. We simply allow the cells to flow into a chamber. As they touch on one surface, we move the cantilever over and lower it down onto the cell. The cell will then spread on both surfaces if they're coated with ECM. So this is an example where the extracellular matrix for these epithelial cells is important. And so if we coat both the bottom surface and the top surface with the same proteins, and we move the cantilever on top of the cell, it will spread onto both surfaces and then pull. Okay, so here's a side view image of what it looks like for this osteosarcoma cell as it pulls on a cantilever. So the cantilever is on the top. There's a substrate right here, and you can see a bit of a reflection of the fluorescence below. So you're looking at the actin inside of this cell as it pulls on these two surfaces. As you can see that over this about 5-minute movie, it's quite a dynamic rearrangement of the actin cytoskeleton as it's pulling on this structure. So being able to quantify things about the forces that are generated allows us to say something about how much shape change, quantify forces and shape change, allows us to say something about how this cell responds to its physical microenvironment. So we can measure things like the cell length, how much force it's generating, and how does it change shape over time. So this is, again, just using AFM, but in a slightly different configuration that allows you to isolate specific quantities. So now let's get to platelets. So does anybody know how platelets are formed? They're actually formed in a really cool way. So they're formed, I'll show you this movie, from megakaryocytes, which are the precursor cell. And the megakaryocyte basically fractures. It beads off into lots of smaller cells, and those individual parts of the cytoplasm are the things that we call platelets. And so one could argue about whether platelets are really cells or just fragments of cells. So that process of fragmentation, all those little individual particles, those are the platelets. And so what those platelets are able to do are critically important at all times, essentially, not just when we have a big wound, but when we have small ruptures in our vasculature over time. Those small ruptures are constantly being repaired and healed and covered by platelets. So what's the process? So blood clot formation, you have exposure of collagen due to a rupture in a vessel. Could be a very small one. Fibrin is polymerized, and the clots, the platelets grab onto that fibrin gel and pull it together so as to seal up this wound, close the hole. So that clot contraction process has been studied for years, and a bulk assay for doing that is simply take some platelets, mix them with some fibrinogen, and watch them contract. So this is a very simple experiment to do. You can do it yourselves. Take a little drop of blood and isolate the platelets and see what happens. So I think this is probably my blood. I think that these are the experiments that I think the grad students enjoy particularly when they need to harvest some blood from their advisor. And you can see over the course of a couple hours, the gel contracts. It undergoes this dramatic contraction. And the thing that's doing that contraction is this little particle that came from the megakaryocyte, the platelet. Okay, so it's able to undergo this dramatic change, and it's able to do that at the same time as it's changing the physical properties of its environment. So clot stiffness is an important property in both the ability of a platelet to close the wound, but it's also in a number of clinical conditions. If you have a soft clot, it's more likely to break off and move through the bloodstream and potentially cause a stroke. So having clot integrity is an important thing. And the ability of these platelets to not just pull things together, but pull things together and change the mechanical properties of the clot is one of their key attributes. Now, the studies that have been done so far are bulk studies. There haven't been studies on individual platelets and how much can an individual platelet pull. So we decided to try that same configuration of placing a platelet between a cantilever and a surface and ask, can we quantify how much force is being generated by an individual platelet? Part of the reason why it hasn't been done is it's hard to catch them before they activate. They're really, really ready to go. As soon as they sense a fibrinogen-coated surface, they're going to activate, spread, and try and pull. So it's very difficult to catch them in the act, essentially. So the way we did that was by watching them as they were floating around in solution from the side, using the side view AFM. And then as soon as they came close to a surface, we moved on top of them with an AFM cantilever and held them in place and watched as they contract. Yep.
So given that it's got a solid surface at the bottom and the AFM cantilever, both of which are likely to be stiffer than its usual environment, how similar is the force that you're calculating here to what is really exerted in vivo situations?
So these are going to be maximum forces. So we're actually going to get to exactly that question, which is how do you best emulate the microenvironment? So one way to think about it is that the flexibility of that cantilever is probably the best measure of what the platelet thinks it's pulling against. So in this diagram, it's pulling between two fibers, and in this diagram, it's pulling between the cantilever and the surface. So essentially, the stiffness of the cantilever is the thing that is emulating the microenvironment. And your question, I think, is that really a good measure of what's going on? So actually, in the case of the cantilevers that we used, it's a reasonable estimate for the stiffness of a clot. But I'll show you the two extremes. That is when you prevent it from deflecting at all, which is a very, very stiff environment, or when you make it even softer, what happens? And you'll see that there's an interesting capability of the platelet to sense that environment. Okay, so in these experiments, we look at force generation. It's able to contract. It's able to contract with surprising amount of force. So just to put this in perspective, this is probably about 20 times stronger on a per volume basis than almost any other cell. So these platelets have almost nothing in them but the machinery to do contraction. They release a number of factors to stimulate more coagulation, but they're highly tuned to do contraction.
Machinery to do the contraction, you mean actomyosin?
Yep, actomyosin contraction, exactly. Yep. So again, it's the cytoskeleton, but it's only the bits of the cytoskeleton that do contraction that are involved in this process. So if you disable either of those, it doesn't work.
Does the adhesion to the two surfaces enter in at all in terms of how well it's adhering? And then
it does. So the adhesion to this fibrin network is essential. Otherwise, it's going to, if that's the weakest link, then it's going to pull itself off of these fibers. And so as we conducted these experiments, at the end of them, we can do an experiment in which we pull against just increase the force until there is rupture. And that force was always substantially greater than the average contraction force that we were able to get.
So there's no variability that you know of in what you're measuring there in terms of the adhesion between the two surfaces. So that's kind of a fixed thing.
Well, it's going to depend a lot on the concentration of the fibrinogen at the surface, or sorry, not the fibrinogen, the polymer at the surface. If that coating is non-uniform, that could affect the overall adhesion. But the individual adhesion strength has been measured not in our experiments but in other experiments, and it's the number of receptors times that receptor ligand adhesion strength is always much greater than the contraction force.
Sorry, one more technical question.
Sure.
Integrant, coherent, mix, what?
So primarily integrant.
Thanks.
Okay, so this contraction takes place, you'll notice it takes place over a relatively short time scale. And so this is a cell that is built to contract and does it once. Once it contracts, it's done. So if we allow the force to, allow it to contract, and then we try to release the force. So using an AFM, you have the ability to change the load that's on your sample. And so if you reduce the force on that sample, one might think that it's only just stalled here because it's not strong enough. But if you reduce that force, it doesn't do anything. It doesn't contract anymore. It's done. So it's an interesting structure in that it's a one-shot contraction machine. Now to the question of the microenvironment. Is the stiffness really an important factor? What we found was that if we change the stiffness of the cantilever, that is, we use different stiffness cantilevers, or if we do a case in which we feedback such that the separation between the cantilever and the surface can't change, that the stall force and also the loading rate, the rate at which the force increases, changes. But what that says is that the platelet machinery is sensitive to the stiffness of the clot, and in fact is able to pull harder on a stiffer clot. Now why might we want it to do that? What would be a good thing about pulling harder on a stiffer clot? Well, one other piece, so I don't know the answer, but I'll give you a hypothesis that we have. And that is, if you have a heterogeneous clot, which is most likely, there's probably not a great way of creating a clot that is completely uniform in its density of fibrin or its density of platelets. So you're bound to have different amounts of contraction and different mechanical properties. If you have a platelet that can pull harder on a stiffer clot, you have a greater chance of uniform contraction of that clot despite it being heterogeneous. So that's our thinking for why this might be of interest. So we were able to measure the elasticity in this configuration, about 10 kilopascals, and extensibility. So even though it's contracted, it's able to, it's extensible. It can be stretched without breaking. Okay, so back to this question of why might it be stiffness dependent. So here's the hypothesis. If there is heterogeneity in this clot, if you're going to have a contractile element that's sensitive, I'll pull a little bit. If it's soft, I'll pull a lot. If it's hard, then you get a more uniform contraction of the clot. So we're working on ways to test that very idea. Okay, so the question on microenvironments is exactly one that we've been wrestling with. So AFM is great, but the cantilever stiffness is the cantilever stiffness. If we wanted to change the apparent environment of the platelet or of any other cell, we had to change the cantilever. And that's really a slow way to do work, and that's a kind of difficult, if you want to really understand the way that cells react to changes in their physical environment, or really how the mechanics can regulate the behavior of the cell. And so can we vary stiffness dynamically? And so we thought about this for a little while and decided that with the appropriate feedback mechanism, we ought to be able to do that. So the idea is this. I can pretend that the cantilever is a stiff spring if, as the cell is pulling, I measure how much it's pulling, and I displace the base of the cantilever up by a certain amount. So I can essentially fight the cantilever fight the platelet as it's contracting. From the platelet's perspective, all it knows is that it's hard to pull. And if it's hard to pull, that's like a stiff surface. So it's very much like if you catch a baseball. If you don't give any with your hand, from the baseball's perspective, that's a really hard surface. It's going to hit you and then fall off. If, on the other hand, you move your hand with the ball, from the perspective of the baseball, that's a really soft surface. That seems like, my displacement and my change in force the slope was low. So that's the basic idea here, is can we move the cantilever as a function of how much force is being generated to create different apparent stiffnesses? And so we worked out how to do that. The basic idea is that, as I mentioned, to make something appear stiff, move away as it's pulling, and to make it appear soft, move with it as it's pulling. And then you've got the ability to vary your stiffness over a wide range. And interestingly enough, force clamps, which are routinely done in AFM and optical trap and other type measurements, force clamps are just an extreme in one direction. A force clamp is a very, very soft surface because you have no way of increasing force. You can displace all you want. You don't increase force. And a position clamp, you can pull all you want. You don't change position. That's an infinitely stiff surface. So you can get a full range of stiffnesses from a single cantilever. So we demonstrated that with some control experiments, just using an expanding gel, using a feedback algorithm that adjusted the movement of the cantilever base. We could see that the slopes were different, and these different slopes are different apparent stiffnesses. So what about with a cell? What you can do now that you have an ability to change stiffness dynamically is you can ask, does a cell sense that rapid change in stiffness? So if I've got an environment, if my clot is changing its stiffness rapidly, do I have some way of sensing that? Or is one platelet going to sense one stiffness, and only when it's done and another platelet comes in, can it sense that next stiffness? So we wanted to test that. We test that with initially with fibroblast cells. And what I'm showing you in blue is the step change in stiffness. So these are the stiffnesses that are programmed in to the feedback routine. We're going from 1 times K, which is the cantilever stiffness, to a fifth of that, 0.2, to 5, to 1K, and so on. So we're stepping among different resistances to contraction. And what you notice is that there's a marked change in slope each time we change the apparent stiffness. So we're not changing the length of the cell at that instant. We're not changing the force at that instant. The only thing we're changing is the stiffness. And if you do this, if you zoom in, and if you do this a number of other times at different frequencies, we found that the contraction of these fibroblasts, at least, is sufficiently fast to respond to changes in stiffness that are under a second. So half second, even smaller. So very rapid changes, very rapid sensing of stiffness. Not of force, not of length, but specifically stiffness. Yep.
So I have a question about this, in the sense that here you're having a stiffness change that is only in one direction. That is only the cantilever on top is changing its stiffness. Whereas put the cell in a platelet, the stiffness is going to be altered in different directions. So is there any way you can, say, soften the surface on which it is or something, and see if that kind of alters the dynamic of its...
That's a great suggestion. So that's exactly right. We have one dimension has a certain stiffness, and then the other dimensions has a different stiffness. So it's stiff. The cantilever and the substrate are both stiff compared to the change in stiffness in one dimension. I think these experiments show 2 things. One is that changing the stiffness in one of those directions is sufficient to elicit a response. So it doesn't just listen to the highest stiffness direction. It actually is watching for how stiffness is changing in multiple directions. Second is I think we can address that question you have by changing the substrate, making that a gel rather than a solid substrate, and asking if there's a third stiffness, how does this behavior change? We haven't tried that experiment, but that's a good idea. Okay, so for the last example, I want to go to the actin networks. So we've been talking about whole cells, first white blood cells, and their overall stiffness. We talked about platelets and their ability to contract and how the physical environment adjusts how they respond. I want to go now to specific molecules within the cells that are responsible. Cytoskeletal molecules involved in protrusion of colon cells. So here's that movie again of it running into the surface and responding. And the question is, what's going on at that very leading edge that allows it to know something about the force? If this is really just first order reactions like you learn in chemistry, there's no force term there. There's no way for force to be coupled in to the biochemistry. So how really does that happen? So this is the network. I explained it briefly. filaments polymerize, they branch, and they get capped, and you have to be near a surface in order for branching to take place. And that's how you get these organized, oriented networks that are generating protrusion. Okay, so how do you convert force into biochemistry? And this is one of the key ideas in mechanotransduction. That there's some way that you can go from physics, which we learned, to biology and biochemistry that we're learning. There have been a number of ways in which this seems to be happening. One is at adhesion sites. So adhesion sites can be sensitive to the stresses that are applied to them, either by changing the overall structure or increasing the number of proteins that are present. Early on, it was known that motor molecules will slow down their enzymatic activity, will slow down their walking with force. And in particular, this was for muscle myosin. So you apply a load to a muscle. If you try and lift weights, and you try and lift them really fast, there's going to be a force-velocity relationship to that. Finally, unfolding. If you unfold a molecule, you may be exposing new places for molecules to bind, and that can change the activity of the system. So the problem is that at the leading edge of the cell, none of these are really at work. The first adhesions in crawling cells like this don't occur for until a micron or so back. And so force could be coupled into those adhesions micron back, but at the very leading edge, it's just the network of branched filaments that are present. So is there anything inherent in those that could be sensitive to forces? That's what we want to understand. Okay, so again, we'll use our old friend, the AFM, and see if we can figure out a way of quantifying forces and properties of these networks specifically. So in order to isolate what's due to a focal adhesion, what's due to protein unfolding, what we really need to do is take out those proteins of interest and put them together isolated from the cell. So in experiments that I'll show you now, we're doing reconstituted experiments. So these reconstitutions are a process of identifying the particular proteins that are important for a process, purifying them, and then recombining them in a physiologically relevant way. And so the way we've tried to emulate what's going on at the leading edge of a cell is to have soluble proteins in solution here. So the actin, the branching protein, the capping protein, cycling proteins in solution here. And then to have on the cantilever surface, to have this protein that restricts the binding of the branching only to near the surface. So we only have one control, one thing that keeps this from being a random polymerizing network everywhere, and that is putting what's known as a nucleation promoting factor on the surface. And that's just like what the cell does. It keeps that nucleation promoting factor on the cell surface. Okay, so we do AFM experiments with this purified system. What you're looking at in the bright channel there is labeled actin. So as the actin assembles in this network, it's assembling at the cantilever surface, you can see that it pushes the surface down. Or in this case, we have a force clamp in place, so we're not letting the force increase, and we're simply allowing the network to displace at a constant force. And so what this is showing is that with a few proteins mixed together, you can recapitulate the growth of an actin network. Not only that, but you can show that it generates force. It's generating a controlled force underneath this cantilever. So now that we have it in a force microscope, we can do a range of experiments with it. We can look at its elasticity, and we did this a couple years ago to show that the elasticity is quite complex. There's a nonlinearity to both the stress stiffening, and there's a softening behavior that we attributed to buckling of individual filaments. So the elasticity of these networks is unique, and the buckling process results in a reversible stress stiffening. That is reversible in which if you let the stress off, the network recovers its elasticity, and you can go right back to its original elasticity. So other than the mechanical properties, it's really the dynamic properties I want to tell you about. So a way to characterize how dynamic networks work, or how motor molecules work, is to measure a force velocity. That's kind of like, for your car, it's like a torque RPM curve. You want to know how many RPM do I need to go to in order to get a certain torque. Well, it's similar here. How much force can be resisted for a given growth rate. And so to measure that, you can do that easily with a force microscope like this. Simply allow the network to grow. You're measuring rate of displacement, and you're also measuring deflection, which gives you force. So you've got force and velocity directly from a growth curve. And so if you take the growth curve, it looks something like this, and you plot it as a function of velocity and force. What this is saying is that at some point, if I push hard enough, I can stall the growth. This network can't keep assembling if I push hard enough. But if I don't push quite as hard, I'll go faster. And over some range, that's about half of this force-velocity relationship, I get a constant growth rate. That is, I'm growing at a constant growth rate. I'm adapting to this force. I'm not slowing down. So what could be responsible for that? Well, our hypothesis is that what's happening at these branched actin networks is that the application of a force is changing the density of the network. And if I nucleate new filaments, if I create new pushing proteins, what I'm doing is I'm spreading the load over more force-generating polymers, and I'm able to handle a greater load. So that's the idea, that as I increase load at the edge of this growing network, I'm actually nucleating new filaments. Okay, so we've recently seen through a range of experiments that the curvature of the filament affects the branching behavior, and this curvature leads us to believe that when these networks are under load, they're more likely to branch on a surface that generates a new filament towards that surface. So I'll give you a diagram here. So here's the data. If I've got a filament and it's bent that way, I see a greater chance of forming a branch on the outside than on the inside. So this is a clever mechanism for a single filament to be a sensing element, to be a force sensing element, and to bias the growth of new filaments in a certain direction. Okay, so I think I've got to finish up here. So here's the model for what we think is going on as the filaments are being deflected by a surface, whether it's a cell that's crawling into a surface or whether it's a cantilever surface. Filament bending is actually coupled to the branching process, and that coupling leads it to generate more branches in the direction of the load. So again, the idea that force feedback resides within these proteins, within these cytoskeletal proteins, doesn't require the nucleus, doesn't require you to ask questions of the nucleus or transcribe more proteins. This ability to sense and respond is all resident in a small set of proteins. Okay, so I think I will end with one more movie, and that is, does this make any sense? Well, we can do a quick experiment to see if the presence of a force changes the amount of actin that's present at the leading edge. And so this is one of those experiments where I thought, it's not even worth doing. We shouldn't try it, but the students did anyway, and it did work. So what I'm showing here is one of these fish keratocyte cells, and it's running into a glass pipette that's put in front of it. So if we want to know what happens when cells run into objects, what the response of the cytoskeleton is, the simplest way is to transiently put an object there and then figure out how much actin is there. In order to do that, in order for the disassembly process not to take over, we need to freeze the actin, we need to freeze the cell instantaneously. So what you're seeing, that little white flash, is a fixative, glutaraldehyde, that gets washed over the cells and suddenly freezes them in place. Okay, so that's the process. They run into this glass pipette, and then they're frozen in place. And if we then do a phalloidin labeling of the actin, you can see that there's this line of increased density at the leading edge. So it seems like the idea that increased density occurs due to mechanical interactions seems to be true in at least these cells. So I think I'll end there and be happy to answer any questions.
Love the in vitro force generation from actin only. That's the first time I've seen anything like that. That's really cool. Have you? I was seeing. Am I right that we're looking at a few tens of nanonewtons for how much surface area?
For the networks are growing over about a 10 micron square, about 10 microns on edge. So it works out to be something like a kilopascal.
Okay. Have you then tried to see if that comes anywhere near mimicking, say, what Listeria does, push that volume through whatever, however viscous the cytosol of those human cells are?
Yeah, great question. So the changing viscosity has not been able to reach the stall forces that we're seeing here. Right. So it seems like the actin network is very able to move through a wide range of different cytoplasmic viscosities. So to check on that one. A second way to quantify it and to figure out if this range of forces is even realistic, or range of pressures is realistic, is to look at how much force the leading edge of a crawling cell generates. So not taking the proteins out, but look at them in the cell. That was done by a different lab, by the Radmacher lab, and they ended up with a force or a pressure that's almost identical to what we see with proteins alone. So there's some connection between those two. Very nice. Nice shirt,
oh, thank you. You mentioned that the cells use different patterns of actin polymerization.
That's right.
And one of those is the stress fibers. Do you know if this specific formation is induced by some sort of stress force applied to the cells?
Yeah, very good question. So actually, you can kind of make out stress fibers in the back of the cell, and they need to form between adhesion sites. And so there's a process that starts with adhesion and then links multiple fibers in the cortical cytoskeleton together through alpha-actinin as well as myosin, and you start to assemble these stress fibers. Now, the actual process for how they get triggered is not very well understood. The proteins that are involved in them are starting to become better understood, but there's still a lot of work to be done to figure out how does the link get formed? Why is the stress fiber between that location and that location? It's certainly true that different elasticity substrates will give rise to different numbers and different amounts of stress fibers. So there are external signals that influence how stress fibers are organized, but the exact mechanism for their assembly is not well studied.
So you said that if you, like when you were talking about the actin growth, like if you push hard enough, like apply greater, large enough force, and you just stall growth. Like, so have you all like tried to do like a definitely like tumor suppression or anything like that, like trying to revive poison definitely?
Yeah, that's an interesting idea. We haven't tried it for very long. I mean, once we stalled it, we had assumed that it would stop growing, but that's an interesting thought about how you could control motility. I think one of the challenges is going to be you want to, if we were to push on the side of the cell, it wouldn't care as much. It would just keep going. And so being able to stop it in the right way is probably the biggest challenge in using some mechanism like that to slow growth of a tissue. The broader question is just how do the complex set of forces that are present during tissue morphogenesis, how do those guide growth, or are those ignored during growth? And the same would be true for tumor cells. Do they simply ignore the loads that are put on them, or are they really sensitive to it? So don't know. I don't know is the short answer.
You mentioned that leukemia is called the platelet or white blood cell adhesion field. And many diseases is about the cell adhesion field. So have you ever used that AFM to detect the patient's maybe monocytes or white blood cells adhesion ability to relate to the mortality or the outcome of the patient?
So you're right about adhesion being a very important process and involved in a number of diseases. We have used AFM. I showed one image of two cells together. The AFM had a white blood cell attached, and then we had an endothelial cell on the surface, and we were looking at attachment forces between the two. We've done that not for a particular disease, so that's a good idea for experiments that would let you look for abnormalities in adhesions. But we haven't done experiments like that.
I'm thinking that since cells can respond to the physical microenvironment, in other words, mechanical properties may regulate cell behavior such as migration. So I'm thinking if these physical parameters have been used to evaluate the influence on the stem cell differentiation.
They have. I think that's one of the most interesting results in the last few years has been the observation that physical cues can take the place of chemical cues in guiding cell fate decisions. So when a stem cell is deciding whether to become cell X or cell Y, its sensing of the physical environment around it will help to guide it in one way or the other. So that has been seen, particularly with mesenchymal stem cells. But more and more cells are appearing sensitive to that. I think the big question is, what's the mechanism? You know, I've pointed out one mechanism that has to do with a particular cytoskeletal network and how individual filaments can sense forces. But what really is the critical steps that are necessary for a cell to sense, I'm in this environment, I want to become that cell? That's a much more complicated question. And I think we're only at the early days of understanding what's involved.
So I was pretty interested with the fact that you can actually do a side view of the fluorescent image, a side view fluorescent image of the cell as you're doing the stress test on it with the AFM. So have you considered, like, since there's got to be something in the interface of the cell between the fiber within the cell and the extracellular matrix, which is likely to be an integrin, have you tried to look at imaging any maybe structural alterations in the integrins as there is this pore-space sensing going on? Because it's likely that integrin is involved in the mechanosensing. So
absolutely.
Look at perhaps. Have you considered doing something like that?
Yeah, absolutely. So we've just set up a system where we can now look at the interface with TURF, total internal reflection fluorescence. So we can get a very good measure of the organization of the adhesions as we're applying controlled loads or different mechanical environments, elasticities to the cells, and figure out, is there reorganization? One interesting question is, if I change the stiffness that the cell is feeling in contraction, does that dramatically alter the organization of the adhesions? Or are the adhesions just set up? And once they're set up, they're going to be there. Maybe they'll be used, maybe they won't be used. So do the adhesions actually dynamically respond? It seems that the actomyosin contraction does, but whether the adhesions do or not is another matter. So yeah, good experiment.
I feel like it's been shown that focal adhesions bulk up as you apply tension to them. So you
Absolutely.
make it bigger. Yeah.
Absolutely. So, and then the question is, do they bulk down? So if you go increase the force of the stiffness and then you go back down, how reversible is that?
There are kinases there at the focal adhesion as well, right? Which can do the
Yep.
signal transduction, which could answer your question. But you're right, early days getting there. Yeah.
Other questions? If not, let's thank Dan once again.
Okay, before you, right, just want to make sure that everyone has the guide, and then thanks. Guidelines for the report that you received, otherwise you have to find extra copies if you have not. The lunch is out there. Those who are going to go to the ESAC, you need to start early, 15-minute walk, looking for the CIT. The students are here, so they will help you with the extra.